हमारे मध्य में उपस्थित हो रहे हैं। आप अपने स्थान पर खड़े होकर सभी अभिनंदन महोदय का अभिनंदन करें। एग्रीकल्चर फॉर टेक्निकल एजुकेशन ऑफ फार्मिंग सोसाइटी एवं वाइटल बायोटेक पब्लिशर्स कोटा के संयुक्त सहयोग से राजकीय महाविद्यालय हिसार के वनस्पति विज्ञान, जंतु विज्ञान एवं जैव प्रौद्योगिकी विभाग द्वारा हाइब्रिड या ऑनलाइन एवं ऑफलाइन मोड में आयोजित हो रही प्रथम एक दिवसीय अंतर्राष्ट्रीय सम्मेलन के इस उद्घाटन सत्र में मैं डॉक्टर मुकेश कुमार अपनी वरिष्ठ सहयोगी डॉक्टर निर्मला राठी मलिक के साथ माननीय प्राचार्य महोदया, आयोजन समिति सहित समस्त महाविद्यालय की ओर से आप सभी का हार्दिक अभिवादन, अभिनंदन एवं स्वागत करता हूँ। राजकीय महाविद्यालय हिसार की यह ज्ञान स्थली विभिन्न साहित्यिक, सांस्कृतिक, सामाजिक एवं वैज्ञानिक कार्यक्रम संगोष्ठियों के आयोजन की समृद्ध श्रृंखला की साक्षी रही है। इसी क्रम में महाविद्यालय द्वारा अत्यंत महत्वपूर्ण एवं प्रासंगिक नेचुरल रिसोर्स एंड एनवायरमेंटल सस्टेनेबिलिटी फॉर फ्यूचर जनरेशंस अर्थात प्राकृतिक संसाधन प्रबंधन एवं भावी पीढ़ियों के लिए पर्यावरणीय संधार्यता विषय पर अंतर्राष्ट्रीय पटल के वैज्ञानिकों, विचारकों, दार्शनिकों एवं शोध अध्येताओं के मध्य मंथन किया जा रहा है। इस अवसर पर हम गौरवान्वित हैं कि ऑफलाइन मोड में हमारे मध्य में इस कार्यक्रम के मुख्य अतिथि गुरु जम्भेश्वर विश्वविद्यालय के माननीय कुलपति एवं वैज्ञानिक के गरिमामयी हस्ती श्री प्रोफेसर नरसिंह जी विश्नोई, इनको हम अपने मध्य में पाकर गौरवान्वित महसूस कर रहे हैं। अब मैं निवेदन करूंगा हमारे इस अंतर्राष्ट्रीय सम्मेलन की संगठिका हमारी प्रेरणा स्रोत माननीय प्राचार्य डॉक्टर दीपमाला लोहान जी से कि वो आए और माननीय मुख्य अतिथि महोदय का वाचिक अभिनंदन करते हुए स्वागत वक्तव्य प्रस्तुत करें। माननीय प्राचार्य महोदय।
Agriculture Form for Technical Education of Farming Society and Biotech Kota प्रकाशन के सौजन्य से राजकीय महाविद्यालय हिसार के बॉटनी, जोलॉजी, बायोटेक्नोलॉजी डिपार्टमेंट द्वारा प्राकृतिक संसाधनों का प्रबंधन और भावी पीढ़ियों के लिए पर्यावरणीय संधारणीयता विषय पर ऑनलाइन एवं ऑफलाइन रूप में आयोजित एक दिवसीय अंतर्राष्ट्रीय सम्मेलन में मुख्य अतिथि के रूप में पधारे प्रोफेसर नरसिंह विश्नोई, कुलपति गुरु जम्भेश्वर विज्ञान एवं प्रौद्योगिकी विश्वविद्यालय हिसार का अभिनंदन सहित स्वागत। इसके साथ ही कार्यक्रम के संयोजक डॉक्टर सतीश वर्मा, विभिन्न विश्वविद्यालयों एवं महाविद्यालयों से पधारे प्राध्यापक, शोध छात्र, मीडिया कर्मी एवं आगंतुक प्रतिभागी, आप सभी का हृदय की गहराइयों से अभिनंदन एवं स्वागत। सृष्टि का संतुलन पर्यावरण पर आश्रित है। प्राकृतिक तत्वों के अति उपभोग से पर्यावरण पर कुप्रभाव पड़ रहा है। भूकंप, बाढ़, पहाड़ी चट्टानों का टूटना, कमी या समाप्ति, भूमि की उपजाऊ शक्ति में कमी, बेमौसम बारिश, कहीं अधिक बारिश तो कहीं अकाल, सर्दी गर्मी की प्रचंडता, खाद्य तत्वों की विपुलता, बढ़ते शारीरिक एवं मानसिक रोग आदि के रूप में इनके दुष्परिणाम हमारे सामने आ रहे हैं। आधुनिक सुविधाओं का उपयोग करते हुए पूर्वजों से प्राप्त इस सुंदर संसार को भावी पीढ़ियों को सौंपना हमारा दायित्व एवं कर्तव्य है। इस महत्वपूर्ण एवं ज्वलंत विषय पर आयोजित यह अंतर्राष्ट्रीय सम्मेलन अत्यंत उपादेय है। इस सम्मेलन के मंथन से उत्पन्न विचार एवं विद्या पर्यावरण संरक्षण में अहम भूमिका निभाई। इस सम्मेलन के उद्घाटन सत्र में पधारे प्रोफेसर नरसिंह बिश्नोई वर्तमान में गुरु जम्बेश्वर विज्ञान एवं प्रौद्योगिकी विश्वविद्यालय हिसार के कुलपति पद को सुशोभित कर रहे हैं। आपका जन्म 1961 को हरियाणा के सिरसा जिले की बॉटनी तहसील में स्थित गांव जांडवाला बिश्नोईयान में हुआ। आप छात्र अवस्था से ही सुयोग्य एवं प्रतिभाशाली रहे हैं। प्रारंभिक शिक्षा के बाद आपने चौधरी चरण सिंह हरियाणा कृषि विश्वविद्यालय हिसार से उत्कृष्ट शैक्षणिक रिकॉर्ड के साथ एमएससी तथा प्लांट फिजियोलॉजी में पीएचडी की गौरवपूर्ण उपलब्धियां प्राप्त की हैं। आपका शोध और शिक्षण के क्षेत्र में लगभग 30 वर्षों का सुदीर्घ अनुभव है। आपने 15 वर्षों से अधिक समय तक गुरु जम्बेश्वर विज्ञान एवं प्रौद्योगिकी विश्वविद्यालय हिसार में पर्यावरण विज्ञान और इंजीनियरिंग के प्रोफेसर के रूप में कार्य किया है। आपने गुरु जम्बेश्वर विज्ञान एवं प्रौद्योगिकी विश्वविद्यालय हिसार में पर्यावरण विज्ञान और इंजीनियरिंग विभाग के विभागाध्यक्ष, डीन ऑफ एमसी एंड ईएस, डीन ऑफ कॉलेज, डीन ऑफ फैकल्टी ऑफ लॉ, डीन ऑफ रिसर्च के महत्वपूर्ण पदों पर सेवाएं प्रदान की हैं। आपने 22 पीएचडी, शोध प्रबंधों और बायो एनर्जी से संबंधित स्वच्छ प्रौद्योगिकियों के विशेषज्ञता के साथ विभिन्न R&D परियोजनाएं पूरी की हैं। आप गुरु जम्बेश्वर विज्ञान एवं प्रौद्योगिकी विश्वविद्यालय हिसार के पर्यावरण और इंजीनियरिंग विभाग में DST FIST कार्यक्रम के अध्यक्ष रहे हैं। आपकी योग्यता एवं शोधपरक उपलब्धियों के कारण अमेरिका की स्टैनफोर्ड यूनिवर्सिटी ने आपको वर्ष 2020, 21 और 22 में विश्व के शीर्ष 2% उत्कृष्ट वैज्ञानिकों की सम्मानित सूची में स्थान दिया। प्रोफेसर बिश्नोई कई पुरस्कारों से सम्मानित भी हुए हैं, जिनमें IIFS द्वारा 2010 में राष्ट्रीय गौरव पुरस्कार, NESA द्वारा 2015 में सर्वश्रेष्ठ वैज्ञानिक पुरस्कार, 2019 में डॉक्टर MA सागरे का सर्वश्रेष्ठ वैज्ञानिक पुरस्कार, 2021 में पर्यावरण विज्ञान में NESA फेलोशिप पुरस्कार से आपको नवाजा गया है। आपकी प्रशासनिक कुशलता भी अत्यंत सराहनीय है। आप 2 मई 2023 से GJU के कुलपति पद के महत्वपूर्ण उत्तरदायित्व का निर्वहन कर रहे हैं। आप युवा छात्रों के लिए प्रेरणा स्रोत हैं। आप जैसे बहुमुखी वैज्ञानिक पृष्ठभूमि के व्यक्तित्व को आज अपने मध्य पाकर हम अत्यंत गौरव का अनुभव कर रहे हैं। इस सम्मेलन के सुचारू आयोजन के लिए कार्यरत डॉक्टर सतीश वर्मा सहित उनकी पूरी टीम साधुवाद की पात्र है। मुझे आशा भी नहीं अपितु पूरा विश्वास है कि इस सम्मेलन से पर्यावरण संरक्षण के विषय में नवीन चिंतन का विकास होगा तथा व्यवहारिक कौशल में वृद्धि होगी। अंत में इस महत्वपूर्ण आयोजन की सफलता की कामना करती हुई मैं अपनी वाणी को विराम देती हूँ।
धन्यवाद महोदया। हम वास्तव में इस प्रथम अंतर्राष्ट्रीय सम्मेलन के कार्यक्रम के आयोजन में उत्साहित है और साथ ही माननीय मुख्य अतिथि महोदय की यहाँ आगमन से गौरवान्वित भी हैं कि उन जैसा व्यक्तित्व हमारे मध्य में विराजमान है। आप सामाजिक दृष्टि से देखा जाए तो आप ऐसे समाज से आते हैं जिस बिश्नोई समाज ने पर्यावरण के विषय में एक नवीन चेतना, एक नवीन क्रांति का आगाज लगभग आज से 500 वर्ष पूर्व बलिदान देकर के किया था। अब मैं इस कार्यक्रम के प्रथम अंतर्राष्ट्रीय सम्मेलन के संयोजक डॉक्टर सतीश वर्मा जी को मंच पर आमंत्रित करना चाहूँगा कि वो आए और इस सम्मेलन की रूपरेखा के विषय में संक्षिप्त प्रकाश डालें। माननीय श्री सतीश वर्मा।
Thank you, Dr. Mukesh ji. A very good morning to all of you present here. I feel privileged to welcome Honorable Chief Guest, great environmentalist and excellent administrator, Professor Narsiram Bishnoi ji, worthy Vice Chancellor, Guru Jambheshwar University of Science and Technology, Hisar, worthy Principal of Government College, Hisar, and Patron of the conference, Dr. Dept ji, Honorable President of AFTES and Vital Biotech Publication and Conference Director, Dr. Jitendra Mehta ji, respected keynote speakers, Dr. Vishwamitra Sharma, who delivered his lecture online on Zoom. He is from Department of Biomedical Sciences and Diabetes, Athens, USA. Dr. Parmesh Kumar ji, Professor of Zoology, Kurukshetra University, Kurukshetra. Dr. Sri Prasad Vista ji, Senior Scientist, NSSRC, Nepal. He will deliver his lecture around at 1 p.m. Dr. Baljeet Singh Saran ji, Principal Scientist and in charge of Biofertilizer Production Technology, CCSHG Hisar, he is just coming. Dr. Krishnendra Singh Namaji, Vice President of AFTES and Conference Coordinator. Dr. Ramesh Arya, Principal, Government College for Women, Hisar, and Government College, Barwala. He has come on special request. Thank you, sir, for coming and gracing the occasion. Organizing Secretary of Conference, Dr. Nirmala Rathi ji, Dr. Sushila Sampan ji, Dr. Kaur Singh ji. It is an honor for the Government College, Hisar. The Department of Botany, Zoology and Biotechnology are organizing the first international conference on natural resource management and environmental sustainability for future generations in collaboration with Agriculture Forum for Technical Education of Farming Society and Vital Biotech Kota. The conference is being organized in hybrid mode. More than 600 participants from India and all over world are joining the conference in online and offline mode. Participants are from diverse fields including academicians, researchers, students and industry personnel also. The main objective of the conference is to provide a forum for national and international experts to emphasize and exchange local and global experiences. In this conference, it has versatile sub-themes and it aims to discuss the current issues like global warming effects, urban disturbances threatened by resources, ethnobotanical importance of indigenous species, aquaculture biodiversity, deforestation, desertification, natural vegetation and sustainability, organic farming, natural farming techniques, phytogeography, geographical disturbances, water resources management and other sub-themes related to the main theme. There will be speakers from academics, the government institutes, national and international organizations that work on the presented will be published in the proceedings of the conference. This international conference is open to all the administrators, corporates, scientists, researchers, scholars, explorers, and non-governmental organizations working in the fields of all related fields of sciences, humanities, and other related sectors. Furthermore, the conference also offers a chance to conduct oral and poster presentations as well as plenary discussions to examine new prospects in multidisciplinary sciences and other disciplines. There are conference oral presentation awards in each technical session running parallel to 5 spots in the college. There will be best poster presentation awards and there will be best oral presentation award to motivate the academicians and researchers. There are different theme related awards and nominations have been invited worldwide, and screening committee has selected the dignitaries analytically and will be awarded today in this conference. Principal of Government College Hisar, Dr. Dept, faculty members of Botany, Zoology and Biotechnology, management committee of the conference, all the staff members of the college, AFTES and Vital Biotech, as well as all the worthy participants who are present here in this auditorium and are joining online all over the world. And I heartily welcome and thanks all of you and AFTES also for giving the total financial assistance in the conference. Thank you, all of you. Thank you.
धन्यवाद सर। संपूर्ण कार्यक्रम की रूपरेखा के ऊपर आपने प्रकाश डालते हुए अपने वक्तव्य को रखा। इस महत्वपूर्ण अवसर पर इस प्रथम अंतर्राष्ट्रीय सम्मेलन के कॉन्फ्रेंस सोविनियर बुक का विमोचन होगा। मेरा मंच पर उपस्थित मंचासीन समस्त महानुभावों से अनुरोध एवं इस कार्यक्रम के संयोजक डॉक्टर सतीश वर्मा जी से अनुरोध कि वो स्मारिका का इस सोविनियर का विमोचन करें।
एक पुस्तक है और ये वाइटल बायोटेक पब्लिकेशन से प्रकाशित हुई है। कुछ ये नहीं पहुंच पाई आज ट्रांसपोर्ट में प्रॉब्लम के कारण, तो हम उसका ऑनलाइन यहाँ एक विमोचन करना चाह रहे हैं। तो मैं अनुरोध करता हूँ नामा जी से कि ई विमोचन ये भी एक इन्वायरमेंट कंजर्वेशन में एक योगदान रहेगा। थैंक यू सर। मेरे साथ डॉक्टर सुशीला सांगवान ऑथर है और डॉक्टर नीलम मंडल है और एक पंचकुला से हैं डॉक्टर रामचंद्र भट्टी। सभी बॉटनिस्ट हैं और ये एथनोबॉटनी की एक बुक है हमारी। इसमें जो प्लांट रिसोर्सेज है उनके क्या ट्रेडिशनल यूज हो सकते हैं हमारे लिए?
सर
एक मिनट फ्रंट पे दिखाना।
प्राकृतिक संसाधनों का यथा आवश्यक प्रयोग, मिनिमम सीमित प्रयोग करते हुए ई बुक के माध्यम से इस पुस्तिका का विमोचन हुआ। अब मैं आमंत्रित करना चाहूँगा माननीय मुख्य अतिथि महोदय को कि वो आए और अपने वक्तव्य से इस सदन को संबोधित करें। माननीय कुलपति महोदय प्रोफेसर श्री रचिराम जी बिश्नोई।
Good morning to one and all present here. It is a matter of great pleasure for me to inaugurate the international conference entitled Natural Resource Management and Environmental Sustainability for Future Generations. On the behalf of Biotech University of Science and Technology, Hisar, I would like to thank Dr. Dept, Principal Government College, Hisar, dignity of the dais and of the dais, and distinguished speakers, delegates, and faculty members and dear students. Further, I extend my heartfelt congratulations to the organizing team of Department of Botany, Zoology, and Biotechnology, and purpose of this conference is to demonstrate our commitment to solve the major environmental issues like preserving the nature and ensure our environment stay healthy. And the main theme of the conference is natural resource management, environmental sustainability. It is not a mere topic of academic interest, it is a call to action. As we all know that our natural resources are limited, yet our demand upon them are increasing, whether it is the air we breathe and whether we drink water and food from forest and life with ourselves, everything is connected. We must understand that all living beings rely on each other and delegate balance that keep us alive. Environmental sustainability जो वर्ड है ये आज के टाइम के अंदर जो है बहुत ही जरूरी है, क्योंकि यूनाइटेड नेशंस सस्टेनेबल डेवलपमेंट ने आज से 2015 के अंदर जो है 169 टारगेट के साथ 17 लक्ष्य फिक्स किए जो सस्टेनेबल जो डेवलपमेंट के लिए है, ताकि हम ऐसी डेवलपमेंट करें साथ ही साथ इकोनॉमिक डेवलपमेंट भी होती है बिना इन्वायरमेंट के खराब करें, ताकि आगे वाली जनरेशन, प्रेजेंट जनरेशन और फ्यूचर जनरेशन की जो इन्वायरमेंट है वो ठीक ठाक रहे। परंतु जब से 2015 से ये कुछ सस्टेनेबिलिटी गोल्स डिसाइड किए उससे लगता है कि इन्वायरमेंट डे बाई डे ठीक होता गया, उससे ज़्यादा खराब होता ही जा रहा है। तो सस्टेनेबिलिटी गोल्स जो है जिसके अंदर 17 में से जो 6 गोल जो है जो इन्वायरमेंट से रिलेटेड है, गोल नंबर 6 है वाटर और सैनिटेशन, और गोल नंबर 13 to 17 जो है क्लाइमेट चेंज, फॉरेस्ट, डिफॉरेस्टेशन एंड बायोडाइवर्सिटी, जो इनको अपनी कॉन्फ्रेंस का टॉपिक है, सारे उसी से रिलेटेड है। तो नेचुरल वाटर रिसोर्सेज का मैनेजमेंट, जैसे आज की कॉन्फ्रेंस का नेचुरल वाटर रिसोर्सेज मैनेजमेंट, सबसे बड़ा नेचुरल वाटर रिसोर्स है वाटर। वाटर की जो बिना वाटर के हम कोई भी है तो रह नहीं सकते जिंदा। वाटर जो है हमारी धरती के ऊपर 70% जो है वाटर से कवर हुई है, परंतु 70% वाटर जो है वो पीने के लायक नहीं है, सैंपल वाटर है। सिर्फ 3% जो वाटर है जो हमारा ड्रिंकिंग एबल वाटर जो पी सकते हैं, जिसके अंदर 1% वाटर है जो हमारे अवेलेबल है एज ए फ्रेश वाटर, और 2% वाटर है वो ऑलरेडी ग्लेशियर के अंदर बर्फ के पूरे फिक्स है। 1% जो ड्रिंकिंग वाटर जो है, जब हम हिमालय के नज़दीक रहते हैं तो इसीलिए हमें ये लगता है कि हमारे पास पीने का पानी बहुत है। तो नहर जो सारी हमारे पास है उसका ड्रिंकिंग पंजाब, हरियाणा और इस एरिया जो है उसके अंदर जो है ड्रिंकिंग वाटर जो हमें नहरों से मिल रहा है, हमें ये लगता है सारे इंडिया के अंदर से ऐसे ही फ्रेश वाटर की जो है सप्लाई होती है, तो ऐसा सारे इंडिया के अंदर नहीं है। और 1% जो वाटर आता है उसका जो कैसे यूज करते हैं, हमारी जो नदियाँ है, नदियों से साफ पानी आ रहा है, पर फिर जो आते ही तो नदियों के अंदर जो है हम सीवरेज से और से जो है एक बड़ी बड़ी शहर के जो सीवरेज है वो सारे उनके गुलाब में डाल देते हैं, इंडस्ट्री का ब्लेंड है वो डाल देते हैं। हम जो 1% जो फ्रेश वाटर है उसको हम जो है खुद ही अपना करते हैं पानी में। और दूसरा जो है नीति योग में भी हम जो पानी का जो इस्तेमाल करते हैं वो भी हमारा तरीका गलत है। चाहे हम इस्तेमाल करने वाले आर ओ का जो पानी है वो जिसमें आर ओ पानी बनाकर पीते हैं, आर ओ का पानी क्यों पीते हैं? आर ओ का पानी पहले हम नेचुरल नहर का पानी पिया करते हैं, तो नहर का पानी हमने खुद ने बुलेट से खराब कर दिया, इसलिए हम आर ओ का पानी तैयार करने कितना पानी वेस्ट होता है वो सारा वेस्ट होता है। दूसरा हमारा फ्लैश का सिस्टम है, फ्लैश के सिस्टम के अंदर भी जो हमारा मूवी सिस्टम गलत है, तो एक बार फ्लैश करके 15 लीटर पानी खराब होता है। तो पानी की वेस्टेज जो है वो नीति योग भी कर रहा है, सारा उसको पानी का ठीक यूज नहीं कर रहे। दूसरा शहर जो है दिनों दिन शहर बनते जा रहे हैं, वाटर बॉडी है वो खत्म होती जा रही है। तो हमारा जो पानी का इंडिया के अंदर जो है 80% पानी की डिमांड जो है वो ग्राउंड वाटर से पूरी हो रही है। तो ग्राउंड वाटर का हम जो है ज़्यादा ही है और एक्सप्लॉयटेशन हो रही है। 62% हमने ऑलरेडी इसकी और एक्सप्लॉयटेशन ग्राउंड वाटर की हम कर चुके हैं। और हमारे पास में ग्राउंड वाटर को रिचार्ज करने के लिए क्या थी? वाटर बॉडीज थी। तो वाटर बॉडी जो है शहरों के अंदर आजकल पहले हुआ करती थी, अब शहरों के अंदर बिल्डिंग पड़ेगी, वाटर बॉडीज कहीं खत्म होगी। तो वाटर चार्ज पानी को ग्राउंड वाटर को चार्ज करने के लिए रिचार्ज करने के लिए वाटर बॉडीज बहुत जरूरी है, वो दिनों दिन खत्म होती जा रही है। वेटलैंड की वो भी खत्म होती जा रही है। और दो रिवर्स है, सेंट्रल पोल्यूशन कंट्रोल बोर्ड में काफी इंडिया के अंदर 275 जो रिवर्स है जिसके अंदर पानी पीने के लायक नहीं है। तो रिवर्स का क्या है? वाटर बॉडी हमने खत्म कर दी, वेटलैंड हमने खत्म कर दी। तो आने वाले टाइम के अंदर ऐसा ही आप रहा तो पानी का एक बड़ी समस्या। तो दूसरा जो हमें इसके लिए क्या करना चाहिए, तो हमारे हाथ में हमें रेन वाटर की जो है उसकी हार्वेस्टिंग करनी चाहिए ताकि ग्राउंड वाटर का रिचार्ज होना चाहिए। और दूसरा जब भी हम पानी जितना भी सेव कर सके हमें सेव करना चाहिए। जैसे हम टूथपेस्ट करते हैं तो टूथ चला देते हैं, उसको बंद रखनी चाहिए। हम जितना मिनिमाइज हो सकता है हम पानी की वेस्टेज को मिनिमाइज करें। और दूसरा उसके बाद में जो मेन प्रॉब्लम जो आ रही है क्लाइमेट चेंज। क्लाइमेट चेंज जो आज के टाइम में बहुत ही बड़ी समस्या है। क्लाइमेट चेंज की वजह से क्या हो रहा है? बारिश के टाइम बारिश नहीं हो रही, गर्मी के टाइम गर्मी नहीं हो रही, सर्दी के टाइम सर्दी नहीं हो रही, सारी चीज़ें चेंज हो रही है। अब पीछे एक हफ्ते पहले जो है पाकिस्तान में यूनाइटेड अरब इमारात में और में कई कंट्रीज में बारिश इतनी ज़्यादा आ गई जो पहले कभी नहीं हुई, जबकि बारिश के टाइम में बारिश नहीं होती। ये सारी क्लाइमेट चेंज की वजह से है। क्लाइमेट चेंज का मेन कारण क्या है? ग्रीन हाउस गैसेज। ग्रीन हाउस गैसेज कहाँ से आती है? ग्रीन हाउस गैसेज जो है हमारी मेन जो है व्हीकल्स है उनसे आती है। फिर एग्रीकल्चर जो वेस्ट है वो हम जलाते हैं उनसे आती है। सॉलिड वेस्ट जो है हम सारे साल जलाते रहते हैं उसकी वजह से आती है। तो पेडिस्ट्रा जो है जिस तरह एग्रीकल्चर वेस्ट है जिसको हम जलाते हैं, तो इंडिया के अंदर जो है पंजाब और हरियाणा दोनों 30 मिलियन टन जो है 62 मिलियन टन पेडिस्ट्रा जो है 30 मिलियन टन पेडिस्ट्रा जो प्रोड्यूस करता है पंजाब और हरियाणा दोनों, जिसके अंदर जो है 18 मिलियन टन का तो हम उसका एज ए फ्यूल या फीड के अंदर उसको यूज कर लेते हैं। उसका 12 मिलियन टन जो पेडिस्ट्रा जो है वो हम उसको जलाते हैं। पेडिस्ट्रा को जब हम जलाएंगे तो सारी जो है कार्बन डाइऑक्साइड, नाइट्रस ऑक्साइड, सल्फर डाइऑक्साइड, ग्रीन हाउस गैस निकलती है। 1 टन जब हम पेडिस्ट्रा जलाते हैं, 1 टन पेडिस्ट्रा से 1460 किलोग्राम तो कार्बन डाइऑक्साइड निकलती है, 60 किलोग्राम कार्बन मोनोऑक्साइड निकलती है, 2 किलोग्राम जो है नाइट्रस ऑक्साइड, सल्फर डाइऑक्साइड निकलती है और 3 किलो सस्पेंडेड पार्टिकुलेट निकलते हैं और 2 क्विंटल के करीब जो है राख निकलती है। जब 1 टन से अगर इतना जमता है तो 9 मिलियन टन जो है पंजाब और हरियाणा की जो पराली जलाई जाती है तो उससे कितनी ग्रीन हाउस गैसें निकलती है उसका हम अंदाजा लगा सकते हैं। मैं खुद जो है हमारी मेरी लैब के अंदर जो है एयर पॉल्यूशन के ऊपर हरियाणा की स्टडी की थी। हमारे हरियाणा के अंदर जो है आजकल सारी जो सिटीज़ के अंदर जो है हमने इन्वायरमेंट कंट्रोल करने के सारे इंस्ट्रूमेंट लगाए हुए हैं। तो हमारा जो हरियाणा का डेटा है, हम सारे साल जो है एम्बिएंट एयर क्वालिटी जो है 100 उसका है, जो 100 से कम होनी चाहिए मतलब ये। और हमारा हरियाणा का जो एयर क्वालिटी इंडेक्स जो है सारे साल 100 से ज़्यादा रहता है। इसका मतलब क्या है कि हम सारे साल जो है हम जो जहरीली गैस हो गए वायुमंडल के अंदर हम सांस लेते हैं, परंतु इसका हमें फील कब होता है? जब ये अक्टूबर नवंबर दिसंबर में होता है, पहले नहीं फील होता। तो पहले क्या होता है, पोल्यूशन तो पहले भी होता है, और उस टाइम में क्या है कि टेम्परेचर ज़्यादा होता है तो जो भी पोल्यूशन होता है ऊपर चला जाता है, डाइल्यूशन हो जाता है, फील नहीं होता। सर्दियों के टाइम में जब अक्टूबर के अंदर टेम्परेचर डाउन हो जाता है, उस टाइम में टेम्परेचर की इन्वर्जन की वजह से जो पोल्यूटेड गैसेज है, सस्पेंडेड पार्टिकुलेट है, ये ऊपर नहीं जाते और वो नीचे रहते हैं। नीचे रहने की वजह से जो है ना सारे ये स्मोक जो मॉइस्चर हो गया, स्मोक है तो स्मोक बन जाता है, जिसकी वजह से प्रॉब्लम आती है। सारी क्रांति की वजह से प्रॉब्लम है, वो प्रॉब्लम नहीं है, प्रॉब्लम हमारी सारे साल रहती है। वो टेम्परेचर कम होने की वजह से क्लाइमेट मिल रहती है और 3-4 महीने जो है हमें बड़ी भारी दिक्कत रहती है। और दिक्कत हमें सारे साल ही होती है। वो जो अपने अस्थमा के और के जो मरीज है उनको पता रहता है 60 साल में कितनी प्रॉब्लम होती है। ये सारी हमारी खुद की देन है जो हम इसके लिए करती जाती है। इसके साथ ही साथ दूसरी प्रॉब्लम बहुत बड़ी प्रॉब्लम बढ़ती जा रही है सॉलिड वेस्ट में। सेंट्रल पोल्यूशन कंट्रोल बोर्ड जो कहता है कि एक आदमी डेली 620 मिलीग्राम जो है सॉलिड वेस्ट प्रोड्यूस कर रहा है। एवरेज इंडिया के अंदर एक आदमी 420 ग्राम सॉलिड वेस्ट प्रोड्यूस कर रहा है। और टोटल जो है हमारा इंडिया के अंदर जो है 62 मिलियन टन एनुअली सॉलिड वेस्ट प्रोड्यूस हो रहा है। और जिसका 30% सॉलिड वेस्ट तो हम उसको मतलब 70% हम कलेक्ट कर रहे हैं और 30% सॉलिड वेस्ट है जो बड़ी नदियों के अंदर ओपनली चलाते हैं, हम उसको इकट्ठा ही नहीं करते उसको सॉलिड वेस्ट। और 70% जो है हम इकट्ठा कर रहे हैं, उसका 12 मिलियन टन है, उसको ट्रीटमेंट होती है और उसके बाद में तो 30 मिलियन टन जो है उसको हम लैंडफिल में डाल देते हैं। और जो आजकल किसी भी शहर में जाते हैं, आजकल पहाड़ बने हुए हैं सारे सॉलिड वेस्ट के, ये सारा लैंडफिल में हो जाता है। और उसके अंदर जो लैंडफिल से क्या है लीचेज होता है वो ग्राउंड वाटर में जाते हैं और ग्राउंड वाटर को भी कंटेमिनेट करते हैं। हमारी आजकल सारी डिमांड है ग्राउंड वाटर से ही वाटर की कमी पूरी करते हैं। तो एक तो सॉलिड वेस्ट को सारा इकट्ठा करके वो प्रॉब्लम, दूसरी वो ग्राउंड वाटर को खराब करता है। और एक और साथ ही साथ में जो सॉलिड वेस्ट जो है सारा साल हमारा चलता रहता है, तो चलता रहता है तो उसमें सारे साल उसकी पोल्यूशन जो है होती रहती है सॉलिड वेस्ट की। और जो सॉलिड वेस्ट के अंदर जो प्लास्टिक है और दूसरी चीज़ें भी है वो भी चलती रहती है साथ में। और ये चलती है जिसमें बहुत ही जो हार्मफुल जो कंपाउंड्स है वो चलते हैं, उनकी वजह से जो है कार्बन टेट्राक्लोराइड, क्लोरीन, नाइट्राइड और जो पॉलीसाइक्लिक एरोमेटिक हाइड्रोकार्बन, डाइऑक्सिन, फ्लोरीन, इस टाइप के कंपाउंड जो है जहरीले कंपाउंड धुएं में निकलते हैं जो कि हाईली कार्सिनोजेनिक कंपाउंड है। तो आजकल जो कैंसर की रोग की बीमारियाँ बढ़ रही है सारी इन्हीं कारण से बढ़ रही है, तो जब उन्हीं इन्वायरमेंट में रहते हैं जहाँ ये केमिकल निकल रही है। दूसरा जो है हम पेडिस्टार को जलाते हैं, तो जलाते हैं तो उसकी वजह से क्या होता है कि जमीन की जो ऊपर लेयर जो है वो सारी जल जाती है, तो उसके अंदर जितने भी माइक्रो ऑर्गेनिज्म है वो सारे मर जाते हैं। उसके बाद में हमारी एक तो उसकी वाटर होल्डिंग की कैपेसिटी जो है स्ट्रेंथ कम हो जाती है, वाटर होल्डिंग की कैपेसिटी कम हो जाती है। उसके लिए फिर हम क्या करते हैं, फर्टिलाइजर पेस्टिसाइड को नहीं लेने के लिए यूरिया फर्टिलाइजर भी डालेंगे, फिर पेस्टिसाइड इंसेक्टिसाइड भी डालेंगे। और वो इंसेक्टिसाइड पेस्टिसाइड का क्या होगा, उसको बर्ड्स दूसरे जो खाते हैं बायोडाइवर्सिटी जितनी हमारी वो खत्म होती जा रही है। तो पहला जब हम खेत में इरीगेशन क्या करते हैं, वो पानी दे दिया उसके बाद हल चलाया करते हैं, तो उसमें अलग अलग टाइप के बर्ड जाया करते हैं, चिटोड़ी है दूसरी और चीज़ें बर्ड जाया करती है, तो उसे वो सारे जो अपने सूंडी और दूसरे उनको खा जाया करती थी। आजकल वो सारे खत्म हो गए तो बीमारियाँ ज़्यादा होगी, इसलिए पेस्टिसाइड इंसेक्टिसाइड डालते हैं, उनका सारा फूड चेन के अंदर एंट्री होगी, वो सारा बीमारी का कारण बनेगा। और दूसरी एक और प्रॉब्लम जो आजकल जो 99 के बाद में नया एक टाइप का पोल्यूशन आ गया जो है प्लास्टिक पोल्यूशन। 1970 से लेके 99 तक जो पोल्यूशन था वो कम पॉलीथीन का पोल्यूशन था, जिसको हम नहीं हो जाता था। तो 99 के बाद में पॉलीथीन का बहुत ही ज़्यादा यूज होने लग गया और पिछले 20 साल के अंदर इतना हो रहा है कि इंडिया के अंदर जो है 40 मिलियन टन जो है प्लास्टिक पॉल्यूशन होता है, और जिसके अंदर प्लास्टिक ऐसा है कंपाउंड नॉन बायोडिग्रेडेबल है, जहाँ भी ये फैक्ट्री में जाएगा, समुद्र में जाएगा, रिवर में जाएगा, वहाँ पे ये सालों साल पड़ा रहता है, कोई डिग्रेड नहीं होता। और इसमें जो पॉलीथीन है, इसमें छोटे छोटे पार्टिकल जो है वो सांस के द्वारा हमारे शरीर के अंदर निकल जाते हैं, तो वहाँ जाके हमारे डिपोजिट हो जाते हैं और ये एक बीमारी का कारण है। प्लास्टिक की एक नई प्रॉब्लम जो है उभरती हुई आ रही है, एक इलेक्ट्रॉनिक वेस्ट है, ये भी एक टाइप के नए टाइप की प्रॉब्लम जो है नया रूप आगे और ज़्यादा हमारे धारण करेगी। ये सारी जो पोल्यूशन है, जो एक साइड में हमने यूनाइटेड नेशंस सस्टेनेबल डेवलपमेंट ने 17 गोल डिसाइड करे ताकि हमारा भविष्य आगे कैसे हम सस्टेनेबल रख सके। दूसरी साइड पर पोल्यूशन जो है डे बाई डे बढ़ रहे हैं, और उसका भी खुद कारण है, तो हमें जो है इसका यूज करना पड़ेगा। कैसे करना पड़ेगा? जैसे सॉलिड वेस्ट मैनेजमेंट है, उसको हमने क्या करना, इसका गवर्नमेंट है, गवर्नमेंट इतना नहीं कर सकती, इसमें 3R का फार्मूला है, रिड्यूस करो, फिर रिसाइकिल करो, रीयूज करो। रिड्यूस में क्या होगा? सॉलिड वेस्ट को बायोडिग्रेडेबल, नॉन बायोडिग्रेडेबल अपने घर के अंदर सेपरेट करोगे तो 50% तुम्हारा बायोडिग्रेडेबल है, उसको वर्मी कंपोस्टिंग की जा सकती है, सेक्टर में कहीं भी अलग अलग रखा जाएगा उसको, और उसको जो है एज ए फर्टिलाइजर यूज हो सकती है, किचन गार्डनिंग हो सकती है। और दूसरा जो तुम्हारा जो है नॉन बायोडिग्रेडेबल है, उसको सेपरेट में उसमें गिलास है, एल्यूमिनियम फोल्ड है, या दूसरे रिसाइकिलेबल जो है वो अलग किए जा सकते हैं, और रिसाइकिल को रीयूज किया जा सकता है। तो ये एक सेपरेशन का तरीका है, सॉलिड वेस्ट की जो प्रॉब्लम है खुद ही कम करनी पड़ेगी, अदरवाइज ये कम होने वाला नहीं है। दूसरा पॉलीथीन है, पॉलीथीन का हमें कम से कम यूज करना चाहिए, ताकि पॉलीथीन को जब ये मार्केट में जाए हो जाए तो हमें दूसरा कपड़े का या दूसरा टैग लेके जाना चाहिए। और सिंगल यूज पॉलीथीन तो कभी भी यूज नहीं करना चाहिए। पॉलीथीन वो यूज करो जो गवर्नमेंट ने प्रिस्क्राइब कर रखा है, 50 माइक्रोन से 100 माइक्रोन की जो थिकनेस का पॉलीथीन है वो यूज करो, या जो बायोडिग्रेडेबल पॉलीथीन वो यूज करना चाहिए। और जो दूसरा आजकल घरों में मैरिज हो रही है, उसमें प्लास्टिक के कप यूज करते हैं, तो वो हमें नहीं करने चाहिए, सेरेमिक के कप यूज करने चाहिए। तो दूसरा आजकल डेकोरेशन में और प्लास्टिक का बहुत ज़्यादा यूज होने लगा, ये चीज़ें हमें अवॉइड करनी चाहिए, ताकि आने वाली खुद के लिए तो आने वाली जो जनरेशन है उसके लिए जो आदमी भविष्य होगा ठीक हो सके इन्वायरमेंट। तो आज जो कॉन्फ्रेंस है बड़ा रेलेवेंट टॉपिक है, इसके लिए मैं तो होंगे उन्होंने इस कॉन्फ्रेंस का टॉपिक रखा है, इतना बढ़िया रखा, तो बधाई के पात्र है। डिपार्टमेंट ऑफ बॉटनी, जूलॉजी, बायोटेक डिपार्टमेंट जो ऑर्गेनाइज करी, इनको हालांकि धन्यवाद करता हूँ मुझे बुलाया। जय हिंद, जय भारत।
Thank you, sir, for your insightful thoughts. You have given us a good food for thought and stimulated us to think on water conservation. Pesticides affect not only the target animal but also non-target organisms like human beings, honeybees and other useful animals. The upsurge of technology has revolutionized our lives. The ever-coming increasing concern for the world, and we are delighted to have Dr. Parvesh Kumar amongst us. He is a zoology professor in the Institute of Integrated and Honors Studies. We have a good change in the program. We are having an award ceremony in between because this is a shortage of time.
जिन हमारे फैकल्टी मेंबर्स जो अलग अलग कॉलेजेस से इंडिया के अलग अलग हिस्सों से आए हुए हैं, अवार्ड के लिए राइट हैंड साइड से आएंगे और लेफ्ट हैंड साइड से वहाँ लेकर चले जाएंगे एक एक करके, जैसे भी नाम अनाउंस हो। थैंक यू।
The first award goes to Dr. Vipin Bapat. I invite all the delegates over dais to present away the awards. The first award goes to Dr. Vipin Bapat for Best Academician Award. This is from Hisar. Next, this is next Mrs. Jaya Mehta for Best Entrepreneur Award. Next, Dr. Anup Padoli is here in line. We have Dr. Anup Padoli over here. Dr. Karan Singh Gill has been awarded Excellence Teaching Award. Next, Dr. Satish Singla. Next award goes to Dr. Satish Singla. She has 12 startups to 100x. Next is Dr. Satish Singla. He's been awarded Outstanding HOD Award. Next, Dr. Sushila Sangwan. She has been awarded National Award for Best Ecologist. She has authored various books and chapters. Next recipient is Dr. Suresh Kumar. Dr. Suresh Kumar has received National Award for Best Environmentalist. And next, Dr. Ravindra Kumar for Outstanding PhD Thesis Award. Then we have Dr. Rajiv Verma.
इस प्रथम अंतर्राष्ट्रीय सम्मेलन में आने पर इस कार्यक्रम को भव्य गरिमा देने पर एवं अपने वक्तव्य से पर्यावरण विषयक नवीन चेतना हमारे सदन में पहुंचाने पर मैं माननीय प्राचार्य महोदय एवं संयोजक डॉक्टर सतीश वर्मा जी की पूरी टीम की ओर से एवं महाविद्यालय परिवार की ओर से माननीय कुलपति महोदय श्री प्रोफेसर नरसी राम जी बिश्नोई का हृदय की गहराइयों से हार्दिक आभार व्यक्त करता हूँ एवं कृतज्ञता व्यक्त करता हूँ कि जो इन्होंने अपने बहुमूल्य समय में से व्यस्त समय में से थोड़ा सा समय हमें निकालते हुए इस ज्ञान यज्ञ में अपनी आहुति दी एवं पूर्णाहुति के रूप में इस कार्यक्रम को सफल करने का हमें आशीर्वाद प्रदान किया। अब माननीय मुख्य अतिथि को अति आवश्यक कार्यवश यहाँ से जाना होगा, तो हम उनके इस बहुमूल्य समय के लिए जो इन्होंने हमें समय दिया, हम उनके आभारी हैं एवं मैं आयोजन समिति के सदस्यों से अनुरोध करूंगा कि वो उन्हें सम्मान सहित प्राचार्य कार्यालय की ओर अपने गंतव्य की ओर उनका अनुगमन करते हुए उन्हें सानिध्य प्रदान करें। धन्यवाद महोदय।
डॉक्टर कमला मलिक, सॉरी, क्योंकि आई डोंट नो, क्योंकि पहले मिला नहीं हूँ आपसे। हाँ, समान पट्टिका से आपका अभिनंदन करेंगे डॉक्टर राजपाल जी और डॉक्टर श्रीका। बहुत अच्छे रिसर्चर हैं, यूएसए में अभी इसे अवार्ड दिया गया है डॉक्टर आर्य को। डॉक्टर कमला मलिक जी, मैडम प्लीज, शी विल चेयर द सेशन। डॉक्टर श्रीका, और डॉक्टर शुभम लांबा जी। डॉक्टर शुभम लांबा जी प्लीज। डॉक्टर राजपाल, मेरा अनुरोध भी रहेगा सभी स्टाफ मेंबर जो यहाँ कॉलेज के हैं वो कॉन्फ्रेंस हॉल में आए, और बहुत अच्छे लेक्चर हमारे कीनोट स्पीकर हैं डॉक्टर परमेश जी, हरियाणा एग्रीकल्चर यूनिवर्सिटी के हॉस्टल में हम इकट्ठे रहे हैं और वेल नोन जोलॉजिस्ट हैं। बहुत अच्छा इनका इंटरनेशनल प्रेजेंटेशन रहती है, तो थैंक यू सर। और मैं एक ब्रीफ बायोडाटा के लिए अनुरोध करूंगा डॉक्टर निर्मला राठी जी, प्लीज।
Ecology and animal behavior, environmental science, rodent pest management. Dr. Parvesh has authored over 45 research papers in reputed journals and contributed to several book chapters. He has guided 2 PhD scholars and 8 students are pursuing PhD under his guidance. He has 2 research projects funded by UGC, Government of India and Kurukshetra University. He has presented 10 radio talks on biodiversity conservation and rodent pest management. Member of various academic and administrative bodies, member of various professional and social organizations. So we are honored to have you here. His presentation title is From Humans to Preservation, Understanding Bird Diversity for Sustainable Development.
respected chairperson of the session, dignitaries of the dais, respected delegates and young scientists present over here. Good afternoon to all. First of all, I congratulate the entire organizing team for organizing such a wonderful first international conference in this government college. So, from core of my heart, I congratulate the entire team. Secondly, at the same time, I express my heartfelt thanks to the organizing team, the patron of the organizing team, and more specifically Dr. Satish Verma for providing me the opportunity to share some of my findings and the views on biodiversity here. So, as you know, birds, they have fascinated the human being for the centuries due to their beautiful coloration and their sweet songs. And they have been well documented in our tradition, in our culture, and in our history. If we talk about the faithfulness, there is a bird known to be the Sarus crane. It always lives in pair. If one member dies, then the other member stop feeding and sacrifice himself or herself, and this is showing the dedications towards the family. Similarly, today we are talking here about the community harmony, and we are spreading the message वसुदेव कुटुंबकम. हर जगह हमारा यही मैसेज है कि हमें मिलजुल कर रहना चाहिए. But this message is well documented by this community that is known to be the birds. They migrate from one place to the another place. They travel miles of the distance from one place to the another place. Then return back to their home, and this is called as bird migration. And during the bird migrations, they cross so many countries. So they no need of the passport, they no need of the any clarifications or verifications, no visa. Then they return back to their homes. So this is the nice, beautiful example. This is presented by this beautiful community that is known to be the birds. As we know, birds, they are the warm-blooded animals, and they are the animals which are covered by the feathers. Anyhow, due to some technical problems, there is so birds, they are warm-blooded animals. Their body is covered by feathers, which are collectively known to be the plumage. They can fly, they can swim, they can walk, and they lay the eggs and they show the parental care. So there are some interesting facts about the birds that they are found in every ecosystem, whether it is Antarctica, whether it is desert of the Sahara, whether it is tropical rainforest, or it is the large mountains just like the Himalayas. One of the interesting facts is that they migrate so long distance. If we take the example of the Arctic terms, it takes two journey twice a year from north to the south pole. साल में 2 बार नॉर्थ से साउथ पोल के ये चक्कर लगाती है. So this is one of the
they take their journey over the oceans without any stoppage. So this is how they have adapted to their environment, how they have conserved their energy. Then some of the migratory birds, they can migrate at such a height, that is, if we take the example of the bar-headed goat, it is about 20,000 feet above the sea level from where they take their migratory routes. So what is the role of the birds in the ecosystem? They have many ecological roles. They are pollinators. If we take the example of the smallest bird, known to be the hummingbirds, it helps in pollination. They also help in the dispersal of the seeds from one habitat to the another habitat. That is why they maintain the floral diversity of the earth. They may also control over the pests, because some of the birds, they are insectivores, some they are known to be the carnivores. So by this they also maintain the ecosystem balance in the agricultural ecosystem. Some of the birds, as you know, the vultures, they prey upon the dead body of the other animals. So that is why they act as scavengers. Moreover, bird watching is a very fascinating hobby, and this bird watching is also provide, or it adds to the economic of the different countries in the world, and the people, they spend lot of the money or a huge amount on watching the bird activities from one continent to the another continent. The birds, as we have talked in the inaugural sessions, there is climate change. अब उस क्लाइमेट चेंज का क्या है कि हमें कब पता चलता है जब वो मैक्सिमम अपने चेंज पे होता है. But these beautiful birds, they act as an indicator. If we observe these beautiful birds or their activity or their population in a particular environment, they can hint about the change in their habitat or in the environment. That is, we are calling these days, that is known to be the bioindicators. So that is why they are known to be the barometer of this earth or planet ecosystem, because they reflect the changes by their activities, by their population size, by their reproduction, and through which we can manage the ecosystem. Why they are known to be the bioindicator? Because the taxonomy of the bird is well known. Their distribution, behavior, and ecology, they are well studied than any other taxa. But they are mobile. अभी हमने बात की, they can migrate from one area to the another area if they are exposed during the winter migration. Thousands of the birds, they come from other countries in the rural pond or in the other wetland habitats of India. And if we observe, even in Haryana, in the small ponds, rural ponds, they have thousands of the migratory birds. If there is any pollution or there is any deterioration or any habitat characteristics is changed, then you will observe that the number of the species visiting that place, that is decreased, their population number is also decreased. So this is how they reflect the change in the environment, and that this gives an indication to the human being that we go for the restoration of the environment. Next. So this is showing what the beautiful picture of a bird, and this is how their body parts have the entire covering with the help of the feathers, that is known to be the plumage. And these plumage, they are unique. They help in the identification of the bird species, and through these different parts and through their color pattern, their shape of the beak and the size of their feet and what is their feet shape, that can reflect their feeding styles, their living styles. Next. So that is showing what through the shape of their beak we can estimate what is their feeding behavior, whether they are carnivores, whether they are granivores, or whether they are frugivores, or whether they are wedding animals, whether they are winter-feeding animals. Next. Not only the feet of the birds, they also reflect their lifestyle or their diet, whether they are perching birds, whether they are carnivorous birds, or whether they are swimming birds. So by observing their feet, we can reflect the lifestyle of the birds. Next. So now what is plumage? As already discussed that the body of these birds, it is covered with the help of the feathers, which provide them protection, which provide the loss of the heat from the body, which provide them insulation, and all these together they know to be the plumage. And this plumage is a unique to a particular species. That is their colorations and even arrangement of the different colors within the plumage. It reflects the identification of a particular bird. Suppose this is known to be the picture of a white-breasted kingfisher. So these are the what are feathers. These feathers, they are just extension of the skin of these vertebrates, and they are attached to the bones of these vertebrates, and collectively they are known to be the plumage. So by this, even with the help of the plumage or the feathers individual, we can identify the species of the birds. We are very much familiar. This इसका है क्या होगा उसकी फेदर से हम क्लियर कट इंडिकेशन. अब सेकंड क्या है कि हमारे इन्वायरमेंट में कौन कौन से स्पीसीज है, हमें कई बार बर्ड्स दिखाई नहीं देता, बट हमें उसकी वहाँ फेदर से मिल गई तो हमें क्या पता चलेगा, ये आइडेंटिफिकेशन होगा कि ये बर्ड्स इस एरिया में अपनी एक्टिविटी करता है. नेक्स्ट. This is how their bedding patterns that help in their identification. Next. Now coming to the diversity of the birds at the world level, recently in 2022, it has been estimated there are about 11,000 of the bird species on this Earth planet, and they are belonging to 44 orders, 253 families, 2,384 genera. And there are certain areas in the world which are having very huge diversity of the birds. If we are talking about the Amazon forest, it alone is a home about the 1,300 bird species, which constitute about 15% of the total biodiversity of the world. Now next. If we are coming to the diversity of the India, as you know, is one of the 12 biodiversity countries in the world, and in its 10 biogeographical zones, it is a home of about 1,353 bird species belonging to 26 orders, 111 families, and 432 genera. Next. Now, in the total world, there are 30 sites, 30 areas which has been identified, which are having a very rich biodiversity, and these are known to be the biodiversity hotspots. And in world, up till date, 36 such area has been mapped, and out of these 36 biodiversity hotspots, 4 are located in India, that is Himalaya, Western Ghats, Indo-Burman, and Sudan. Next. So now we come to the another main aspect, that the birds, they are not uniformly distributed in all habitats. There are certain areas which are having very rich diversity of the birds. They are known to be the IP site. So first, at the global level, there is a need to identify these important bird and biodiversity areas known to be the IP site. So the main purpose is to identify such areas, and second point, to monitor these areas over the time, whether the number of species is increasing or whether the number of the species is decreasing. Then another is to protect this biodiversity. If there is any change, we have to take the measure to overcome these adverse environmental factors. So over the world, based upon four criteria क्या है हमारे पास कि वहाँ पर बहुत सारे थ्रेटेड बर्ड स्पीसीज होने चाहिए। दूसरा क्या है कि वहाँ पर एक पर्टिकुलर ऐसे स्पीसीज पाई जाती हैं जिनका डिस्ट्रीब्यूशन रेंज वेरी नैरो है, कोई पर्टिकुलर एरिया के अंदर ही ब्रीड करते हैं, वहीं पर जो है वो पाए जाते हैं। सो दैट इज नोन टू बी द रिस्ट्रिक्टेड रेंज स्पीसीज। और एक पर्टिकुलर बायोजियोग्राफिकल एरिया में पाए जाते हैं, दे आर नोन टू बी द बायो restricted species. And some areas are there, they have a large population of the migratory bird species. These four criteria, they are used to identify or to designate a particular region as a IBA site. So all these four criteria in the bird, 13,600 sites, they have been identified as IBA site, and if we take the geographical area of these IBA sites, they are equal to the 9% of the earth terrestrial ecosystems and 2% of the oceans which are covered by these IBA sites. If we talk about the IBA sites in India, so there is an agency that is known to be the Bombay Natural History Society and Biotech International. They have identified 554 IP sites in India which are having a rich diversity of the birds, and they are monitored and then they are regulated by this global network of the IP sites. And if we talk about the Haryana, अगर हम हरियाणा की बात करें तो there are 6 sites which have been identified as IBA sites. अब इसमें क्या है, बसाई लैंड है, गुड़गांव के अंदर हम जाएंगे जहाँ पर पानी बहुत सारा हमें मिलता है, उस एरिया के अंदर अभी तक जो है 239 स्पीसीज दे हैव बीन आइडेंटिफाइड। इसी तरह से पिंडावास वाइल्ड लाइफ सेंचुरी है, अगर हम जाएं वहाँ पर देखें, और अब तक जो वहाँ पर 265 स्पीसीज ऑफ द बर्ड्स दे हैव बीन आइडेंटिफाइड। कालेसर वाइल्ड लाइफ सेंचुरी, अब इसमें भी कंफ्यूजन आता है कुछ लोगों को कि कई बार हम जो है कालेसर नेशनल पार्क भी कहते हैं, फिर हम कालेसर वाइल्ड लाइफ सेंचुरी भी कहते हैं। नेशनल पार्क इज टोटली प्रोटेक्टेड एरिया फॉर द बायोडाइवर्सिटी, वहाँ पर कोई भी ह्यूमन एक्टिविटी नहीं हो सकती, लेकिन जो वाइल्ड लाइफ सेंचुरी है दैट इज फॉर द वेलफेयर ऑफ द एनिमल्स, वहाँ पे हम जो है कर सकता है। तो जब वहाँ नेशनल पार्क डिक्लेयर हुआ तो उन लोगों को वहाँ के आसपास रहने उसके अंदर एंटर नहीं कर सकते थे, उन्होंने जब एजुकेशन किया तो उन लोगों के लिए कुछ एरिया वो वाइल्ड लाइफ सेंचुरी के रूप में जो है प्रोटेक्टेड है, जहाँ पर वो अपने एनिमल्स को ले जा सकते हैं या ग्रेजिंग को कुछ इस तरह की एक्टिविटीज कैरी कर सकते हैं। तो इसलिए जो ये एरिया आइडेंटिफाइड है एज आईबी साइट दैट इज वाइल्ड लाइफ सेंचुरी। इसी तरह से सुल्तानपुर नेशनल पार्क हम सभी ने सुना है कि इट इज फेमस फॉर द बर्ड्स ऑल ओवर द वर्ल्ड, and there are 320 bird species, wetlands of the Yamuna River, digger wetlands. So these are the 6 areas in Haryana which has been identified as IBS. Next. Now, अब हम किस species को ये कहें कि भी ये threatened species है, किस species को कहें कि ये list concern है? So at the international level, there is a union that is known to be the IUCN, International Union for Conservation of the Nature. It has established 5 criteria depending upon population reduction कैसी है species की, उसकी restricted geographical range क्या है, उसका population का size क्या है, वह किस एरिया में वो population restricted है, और उसके एक्सटिंक्ट होने के चांसेस कितने हैं। डिपेंडिंग अपॉन दीज 5 क्राइटेरिया इन द इंक्रीजिंग ऑर्डर ऑफ द थ्रेट्स दे हैव बीन नोन एज थ्रेटेड स्पीशीज। वन इज नोन टू बी द वल्परेबर, If more threats are there, then we get to the higher category, that is going to be the endangered. And then if there are more threats, more population decline, then it gets converted into what ultimately critically endangered. Then what happens after critically endangered? If we are not able to protect their habitat, their population, ultimately what will happen? They become extinct. अब चीता की बात करते हैं हम, where is cheetah in India? Ultimately, all these steps they ultimately leads to what? That is going to be the extinction of the species. So it is the duty of each individual who is present on this earth. If we are utilizing the resources, अगर हम अर्थ के इतने resources यूज कर रहे हैं, तो equally शेयर जो other organism इस अर्थ पे हैं, उनका भी उतना शेयर उनको मिलना चाहिए. So that is why there should be optimum utilization of the resources. Next, if we talk about the world level, 230 species they are critical endangered, 423 species they are in endangered category, and for 755 they come as the vulnerable species. इन 3 species को हम basically definition में threatened species काउंट करते हैं, and this comes to 11,000 से 11,000 में से 1409 है आज के लिए. They are known to be the threatened species. But for the broader understandings, there also another category that is near threatened. Near threatened वो है जिनकी तरफ अगर ध्यान नहीं दिए तो जल्दी ही they get converted into the higher category, that is threatened species. So over the world there are 1,002 species which are considered to be near threatened. अगर हम ये sum कर लेते हैं, this sum is 2,411 species out of 11,000, and it comes to be 12.8. और एक मोटे उसमें हम कहें वर्ष में कहें तो every 8 species of the world in the world is known to be the threatened species. If we talk about the India में क्या है? There are 82 bird species. अगर हम इसको कैलकुलेट करें, हम 1300 के करीब जो हमारे बर्ड स्पीसीज पाई जाती हैं, so it is on higher side, that is around 14%. Next, these are the top 10 countries which are having threatened bird species. If we see towards the picture, India comes at the 6th rank after the Indonesia, Brazil, Colombia, Peru, and China, because India provides habitat to about 182 threatened species. So that is why we are having a very rich diversity of the birds in India. Next. Now, again there is at the global level agency that is going to be the IUCN. By taking the data over a long period, they define that the population of this species is declining, it is increasing, it is decreasing, or it is stable. So 49% of the species, you may say that 50% of the total bird species, they are having declining population trends. Then 38% they are having stable bird population. 6% are such species whose number is going to increase, and 6% they are species whose population trend is not known. We cannot say whether it is stable because data is not sufficient to have a conclusion whether their population is increasing or their population is decreasing. So if we come to the level of the India, 62% forest birds, they are showing the declining trend, whereas 59 they सर्फ के अंदर जो पाए जाते हैं birds, they are again showing the declining trend, and 47% of the wetland bird species हम उनको कहते हैं which are totally dependent upon the water bodies for their survival, they are not to be the wetland birds. So bird migration, as we move from one country to the another country for bird economic growth or for the luxury life. So the birds, they have already established this habit, but उनके अंदर इतनी लड़ाई या वो नहीं है. They have adopted it peacefully. If in one region where they breed, there is very cold winter, they migrate to the India. यहाँ पे आएंगे, खाना खाएंगे, फिर उनका ब्रीडिंग सीजन हो जाएगा. यहाँ टेम्परेचर हाई इंक्रीज होगा, they will return back to their homes. So that is why they are the best example of वसुधैव कुटुम्बकम. कि जब हमें जरूरत हो तो हम एक दूसरे के साथ अपने रिसोर्सेज को शेयर करें. और कभी तक कोई ऐसी हमारे जो लोकल बर्ड्स जो थ्रू आउट द ईयर हमें पाए जाते हैं, हम उनको रेजिडेंट बर्ड्स करते हैं. उनको कोई कोर्ट केस नहीं करते, वो कोई ऐसी लड़ाई झगड़ा या प्रोटेस्ट नहीं कर सकते या करते हैं कि बाहर से ये आगे हमारा क्या होगा. दैट मीन्स इफ वी ऑप्टिमाइज our demands, then we can live peacefully on this earth and we can maintain the ecosystem for the future generations. So there are 9 flyways. Flyways, these are the regions from which our birds move during one season to the another and then return back. These maps, they are going to be the flyways. So India, it comes on the flyways that is known to be the Central Asian flyway. So in India, there are about 370 bird species which migrate every year through this route that is known to be the Central Asian flyway. If we talk about the Central Asian flyway, it starts from where? Siberia in Russia, and then it crosses and reaches to the South Asia, particularly Maldives. And in this route there is boundaries for these bird crosses through 30 countries. They breed of no visa, no passport, no customs verification. They easily go from one country to the another country. And out of these 370 bird species, 310 bird species, they are dependent upon the wetlands. So that is why during winter, अगर हम सर्दियों में देखेंगे अपने छोटे से तालाब में भी क्या है, हमें माइग्रेटरी बर्ड स्पीशीज आसानी से दिखाई दे जाते हैं. So these are the main site where you can observe the migratory bird species. So this is a small piece of the work. One of my attempt is to give this message. In the early morning when the Vice Chancellor arrived, there was discussion in the office of the principal कि सर हमें भी जो है PhD गाइड के लिए परमिशन मिले और हम भी जो यहाँ पे PhD के लिए इन्वायरमेंट क्रिएट करना चाहते हैं, तो आपको परमिशन भी मिल जाएगी with the passage of the time. Then next question comes, PhD स्टूडेंट भी आ जाएंगे, तो उनको करवाएंगे क्या? Sometime in the minds it comes, if PhD करवाने के लिए there is a need of a large lab, costly instruments. अब जैसे डॉक्टर प्रजीत बैठे हैं, PCR जी हमें जी वो लाखों में करोड़ों में है, तो सेल फोरेसिस करवाना है. This is why one of my message is that for a good research there is no need of, actually that is, I am not denying these costly instruments are not required, but according to our situation, if we are not having costly instrument, we are not having so much space for the research, with the help of with 2 or 3 small instruments like a camera 50,000 रुपये में आ जाता है, बाइनोकुलर है 10-20 हज़ार में आ जाती है, GPS आजकल कैमरे में अटैच होता है, तो छोटे मोटे इंस्ट्रूमेंट से भी आप जो है एक बढ़िया रिसर्च कर सकते हैं. This is the work of one of my students who work on the bird diversity in the village mode of कुरुक्षेत्र डिस्ट्रिक्ट. उसके कुरुक्षेत्र डिस्ट्रिक्ट के अंदर हम विलेज फोर्स को क्या सोचते हैं कि भाई ये तो पशु हैं उनको नहलाने के काम आते हैं या वहीकर उनको वॉश आउट करने के लिए, बाकी तो इसका कोई नहीं। कभी भी हमने उनको बायोडाइवर्सिटी पॉइंट ऑफ व्यू से दे आर रिच साइट ऑफ बायोडाइवर्सिटी ऐसा हमने अभी तक असेस नहीं किया। सो दिस इज वाज ए स्मॉल स्टडी एंड ही हैज सबमिटेड हिज PhD. So this is just in brief what he has observed there in 5 village pond of Kurukshetra district, and he used what simple the point count method and direct observation methods. Next. And these were the 5 ponds. थाना विलेज है बहुत बड़ा, एक जहाँ पे सबसे बड़ा काफी कुरुक्षेत्र के अंदर पौंड है। यूपी से इट कैन बी ए वाइल्ड लाइफ सेंचुरी फॉर द वेटलैंड पार्क। देन काजपुर जटन है, निवासी है, सुनेड़ी खालसा है। तो these were the small ponds, 10 विलेज ponds, और उसके पास का इंस्ट्रूमेंट था, उसके पास बाइनोकुलर थी, उसके पास कैमरा था और उसके पास एक GPS था। Next. So this is showing the location and the habitat of these 5 village ponds. Next. So during his research, he has identified 171 species. अगर मैं हरियाणा की बात करूं, अभी 530 species they have been reported from Haryana. Out of these 530 species, 171 species he has documented from these rural ponds of the 5 villages, which are belonging to 100 Biotechnology and ultimately 18 orders. In India, there are 26 orders. Out of these 26 orders of the birds, 18 orders of the birds, they have been recorded from this small pond of the Kurukshetra district. So this is showing the checklist in detail, which were the species, what was their status, whether they were resident, whether they were migratory, or whether they were the summer migrants. Next. Next. So this is the checklist, and also showing their month of arrival, if they are migratory birds, in which month they come to this area, and at which month they depart from this area. That is showing their migratory behavior. Next. So this is, that is distribution of the birds species according to the orders, and especially for this with 69 species, it was the most prevalent alpine taxa in this area. Next. So this is showing the different families. There is Anididae. Anididae में क्या आती है? Ducks आती है, bees आती है. And this is the most diverse family, having 19 species. And of these 19 species, 17 species are migratory. जब ये स्टडी की गई, इस 19 species में से 17 species they are migratory. So this is what we can conclude how these small ponds they are rich biodiversity sites. Next. So out of this total 171 species, 59 species they were resident, and 37% were winter migrant, 4% was summer migrant. अगर हम दोनों फिगर को मिला दे 74, 37 plus 4, 41%. So this is what 41% of the total bird species present in this area, they are known to be the migratory species. Next. So then this is reflecting their breeding behavior. Again, what is their role in the ecosystem? That is 54 species they are insectivores. That means they are keeping insect on the insect in this particular area. So they also play an important role in maintaining the ecological balance. Next. So this is how many species they were observed with the declining bird populations. But here one important observation was that there are some species which are having decreasing trend at the global level, but they are having very common population of very abundant population in our wetlands of these small village ponds. So it can be concluded that these small ponds, they are playing an important role in the conservation of the fighters. So out of these 171 species, 6 species they were vulnerable, and around 2 species were vulnerable and 6 species they were threatened species. If you talk totally 8 species, they are having conservation importance at the level of the global. So they are known to be the species which should be given priority for their protection at the global level. So these are the pictures of some of these threatened species reported from this area. Common poacher, black-headed ivy, herbaceous poacher, silver tongue, black-tailed goatweed. And if you explore even in this area, you will find such type of the threatened species in this particular area. Next. So now comes to the threats. First threats क्या है? That is degradation of the habitat or loss of the habitats. कि अगर हम पोर्ट्स को क्या है एग्रीकल्चर एक्सपेंशन के लिए क्या करें, उनके लिए करें, या इन्वेस्टमेंट करें, क्या क्या है वहाँ पे वीट इन्फेस्टेशन है. I showed here you will observe that here mostly village ponds. It has covered the entire surface area so that the birds some ऐसे पक्षी हैं जो ओपन वाटर के अंदर जो है इनहेबिटेट करते हैं, तो उनके लिए क्या है खत्म हो जाता है. Then there is poaching. कुछ लोग जो मीट लवर्स हैं ये डायरेक्टली और इनडायरेक्टली जो है ड्यूरिंग दिस माइग्रेटरी सीजन दे गो फॉर पोचिंग ऑफ दिस डिफरेंट बर्ड स्पीशीज. दैट कि हम कर क्या रहे हैं, फिशरी को बढ़ावा दे रहे हैं, लेकिन ऊपर से क्या है वायर्स हमने लगा दिए. वायर्स लगा दिए तो बर्ड्स वो अपने उस रिसोर्स को यूटिलाइज नहीं कर पाता. तो दैट विल हैपन्स दे शिफ्ट टू द अदर एरिया. और सम टाइम व्हेन यू आर कैचिंग द फिश, उसके साथ क्या है आप जो है बर्ड को भी कैच कर लेते हो. सो दीज आर द वेरियस फैक्टर. क्लाइमेट चेंज हमने ऑलरेडी कि हमने जो फिजिकल एनवायरनमेंट है उसमें प्लांटेशन कम कर दी है, उसके अंदर अर्बनाइजेशन बढ़ गई है. अर्बनाइजेशन बढ़ने से अल्टीमेटली क्या है जो बर्ड्स के लिए ग्रीन एरिया से उनकी कहीं न कहीं एरिया जो है डिक्रीज हो रहे हैं. अल्टीमेटली दीज आर द फैक्टर्स व्हिच आर पोजिंग और सीरियस थ्रेट्स to the bird population. So that is why over the area the number of the threatened species is increasing globally. So these are some of the threats occur in the study area. कि कैसे इंडस्ट्रियल वेस्ट है ये डंपिंग होने से उनकी हैबिटेट को लॉस होता है. कैसे डोमेस्टिक अब क्या होता है गांव में जो जितना भी गंदा पानी है नाली का सारा कहाँ जाता है, जो तालाब में जाता है, तो वहाँ से जाने से क्या है उसका ऑर्गेनिक मैटर जो है इंक्रीज हो जाता है. तो अल्टीमेटली कई पौध आप ऐसे देखेंगे जिसमें एक भी आपको दिखाई नहीं देगा. बिकॉज द क्वालिटी ऑफ द वाटर हैज डिटोरियोरेटेड. एज ए रिजल्ट द बर्ड्स दे हैव शिफ्टेड फ्रॉम दिस हैबिटेट टू द अनदर हैबिटेट. सो दिस आर द थ्रेट्स वर ऑब्जर्व्ड इन द स्टडी. So these are the ways by which we can conserve the biodiversity of the birds. सबसे पहले तो जो अगर हम बात करें कि अवेयरनेस कब आएगी जब हम अपने एरिया की बायोडायवर्सिटी को मैप करेंगे, तभी हमें पता चलेगा कि यहाँ पे माइग्रेटरी स्पीशीज आते हैं, हमारे पास थ्रेटेड स्पीशीज आती हैं. जब तक लोकल लेवल पे हम कोई भी स्टडी नहीं करेंगे, आपको जब हमने इस फील्ड के अंदर रिसर्च स्टडी शुरू की थी, हमने एक छोटे यूनिट से शुरू की थी कुरुक्षेत्र यूनिवर्सिटी का कैंपस. वहाँ से हमने बर्ड की डायवर्सिटी डॉक्यूमेंट की थी और वहाँ पे 92 स्पीशीज ऑफ द बर्ड दे वर लिस्टेड. उसके बाद प्रशासन जागा कि भाई यहाँ पे कितने स्पीशीज ऑफ बर्ड्स हैं, उसमें भी क्या है हमारे थ्रेटेड स्पीशीज है. वहाँ पे क्या हो रहा था हॉरिजॉन्टल एक्सपेंशन ऑफ द बिल्डिंग्स. उसके बाद उन्होंने 30 एकड़ लैंड जो है that was dedicated for the welfare of the wildlife. अब आप लोगों के आसपास कितना एरिया है, अगर हम यहाँ का भी एक डॉक्यूमेंट बनाने की यहाँ कौन कौन सी स्पीशीज पाए जाते हैं थ्रू आउट द ईयर, ये एक बहुत अच्छी स्टडी होगी। और इसमें हमें इम्पैक्ट भी अगर 5 साल के बाद दोबारा सर्वे करेंगे कि कितनी पॉपुलेशन इंक्रीज होगी या डिक्रीज होगी। सो दीस आर द एरियाज हियर वी कैन गो फॉर द रिसर्च एंड कैन पब्लिश द पार्टी रिसर्च अगेन कि अगर हम अर्बनाइजेशन इंक्रीज करेंगे लेकिन अर्बन पार्क्स हैं दे आर आल्सो द बायोडाइवर्सिटी साइट्स। अगर हिसार के अंदर कितने अर्बन पार्क्स हैं, वहाँ कौन कौन से बर्ड स्पीशीज पाए जाते हैं और ओवर द ईयर क्या है, अगर ये भी हम मॉनिटरिंग करेंगे तो उससे हमें बर्ड पॉपुलेशन का क्या ट्रेंड है तो ये पता चलेगा। तो मेन माय फोकस इज टू क्रिएट द अवेयरनेस और ये ब्यूटीफुल एनिमल टैक्सा है जिसमें मिनिमम इन्वेस्टमेंट, मैंने कहा कि 2 या 3 इंस्ट्रूमेंट के सिवाय इसमें कुछ जरूरत नहीं है। अच्छे फोटोग्राफ, क्योंकि वो आपको कई बार जो वीडियो के पास भेज देते हो कि ये स्पीशीज तो यहाँ मिलता ही नहीं, तो उसके लिए वैसे कैमरे की भी ज़्यादा जरूरत नहीं है। सो 1-2 आप बुक्स अच्छी सी खरीद लें तो मिनिमम बजट यू कैन हैव वंडरफुल रिसर्च। So I'm thankful to again to the organizer, specifically Dr. Satish Verma, for providing me this opportunity to present my findings. Thank you. Thank you very much. अगर कोई क्वेश्चन है तो कोई बात नहीं है, किसी का भी तो साथी या जिनकी पॉपुलेशन डिक्लाइनिंग। अब मैं आपकी बात का आंसर एक छोटा सा देना चाहता हूँ कि फॉरेस्ट डिपार्टमेंट बहुत ज़्यादा फंड होता है। हमारे हरियाणा के अंदर 8 वाइल्ड लाइफ सेंचुरी जो है उनमें से एक छोटी सी सेंचुरी है चिलचिला वाइल्ड लाइफ सेंचुरी, ठीक है। किसी ने उस पर काम नहीं किया, हमने उस पर काम किया। वो पेपर जनरल ऑफ टेक्सा में आप जाएंगे तो उसमें आपको मिलेगा। अब जब उनके पास बजट आया तो वो हमारे पास आए, सर आप हमारी हेल्प करो। आप आपकी क्या हेल्प करेंगे? हमने अपनी रिकमेंडेशन ऑलरेडी इस पेपर के अंदर मेंशन कर रखी है और उसके बेस पे उन्होंने वहाँ क्योंकि पानी का कोई अरेंजमेंट नहीं था, जो पानी होता था वो ड्रेनेज हो जाती थी एग्रीकल्चर फील्ड से सराउंडिंग है, तो अल्टीमेटली उन्होंने बर्ड वाचिंग प्लान और वहाँ पे पानी जो है अगर माइग्रेटरी बर्ड्स आ रहे हैं, पानी है ही वहाँ पे विंटर के दौरान, तो वो वहाँ पे क्या करेंगे? एक वो सूखा जंगल बन गया था। तो अल्टीमेटली अगर आप इस तरह के एफर्ट्स अभी लेटेस्टली कल ही जो है बायोडाइवर्सिटी बोर्ड के कुछ मेंबर होंगे, उनका सर मैंने आपके पेपर्स देखे हैं, आपका क्या कंट्रीब्यूशन है, तो उनको भी हमने ये कहा हमारा कंट्रीब्यूशन यही है कि जो ये हैबिटेट है, वन ऑफ द हैबिटेट मैं कह रहा हूँ था कि ये जो थाना विलेज पोंड है, ये जो पोंड है, इसका जो साइज है वो 100 एकड़ के करीब टोटल अगर पहले उसको देखें और इट कैन बी ए वाइल्ड लाइफ सेंचुरी। अब अभी तक हम ओरली जो बात करते हैं लेकिन अभी तक किसी ने उसका डाटा जो है पब्लिश नहीं किया है। दैट इज व्हाई आई असाइन्ड इट टू माय स्टूडेंट। उसने 171 बर्ड स्पीशीज जिसमें से 8 स्पीशीज हमने उसमें कैलकुलेट करके दिए कि ये क्या है स्पीशीज है। तो आपका स्टेप बहुत अच्छा है, लेकिन कंटिन्यूसली सर देखिए रिसर्च को हम इसमें सर पेपर में जो कंसर्न जो लोग हैं वो फीड देंगे इसको गवर्नमेंट को डिफरेंट चैनल्स के जैसे ये प्लेटफॉर्म हमें मिलते हैं मीडिया के अंदर जब चीजें जाएंगी। हमारे क्या था कि कुरुक्षेत्र यूनिवर्सिटी में हर वीसी क्या था एक अब वो वर्ड जहाँ यूज नहीं करना चाहिए कि ये होता है कि मैक्सिमम बिल्डिंग होनी चाहिए और जितना वो फॉरेस्ट एरिया था ना वो सारा का सारा काट के जो बिल्डिंग बना रहे थे, तब हमने जब 92 बर्ड स्पीसीज उनको मैप करके दी, तो अल्टीमेटली कुरुक्षेत्र यूनिवर्सिटी की ईसी के अंदर ये पास हुआ कि ये 30 एकड़ लैंड है, तो ये ग्रीन एरिया है, दिस इज फ्रॉम द वेलफेयर ऑफ द वाइल्ड। अब उसको कोई टच नहीं कर पाएगा। तो आप ऐसे एफर्ट्स कर रहे हैं, बहुत अच्छा एफर्ट्स है। और ऐसी बात नहीं, देखो एक दिन में कुछ नहीं होता, बिकॉज इकोलॉजिकल ऐसी स्टडी है कि ओवर द ईयर आप 4-5 साल बाद फिर रिपोर्ट आप जब पब्लिश करोगे कि पहले इतने स्पीशीज थे, अब इतने डिक्लाइन, एक दिन आप भी लोगों की वो बढ़ेगी आप लोगों के भी ऐसे। एनी मोर? थैंक यू। अभी भी ये लीस्ट कंसर्न स्पीसीज है। कुछ हैबिटेट्स में इनका अभी वर्ल्ड स्पैरो डे जो है पीछे सेलिब्रेट किया, इसका नंबर हमारे जो है हैबिटेट को ऑब्जर्व करोगे तो डिक्लाइन हुआ कुछ एरियाज के अंदर। पर्टिकुलरली जैसे एक पहले आया कि भाई ये इलेक्ट्रोमैग्नेटिक वेव के कारण जो है मोबाइल टावर है, लेकिन ऐसा नहीं है, बाद में होती हुई फिर भी अगर आप ओल्ड दिल्ली के अंदर जाओगे तो आपको बहुत ज़्यादा नंबर वहाँ पे मिलेगा पर्टिकुलर रिपोर्ट। वन ऑफ द फैक्टर इज दैट इज चेंज इन द आवर कल्चर एंड द हैबिटेट। अब ये जो हाउस स्पैरो थे ये क्या है, दे आर डोमेस्टिक। डोमेस्टिक मीन्स ये ह्यूमन बीइंग के आसपास ही रहना पसंद करती है, वाइल्ड में नहीं मिलेगी। तो हमारे जो पहले मकान होते थे वो क्या है, कच्चे मकान होते थे या कड़ी वाले मकान होते थे, उसमें कहीं न कहीं छेद होते थे जो इनका फेवरेट ब्रीडिंग साइट है। अब वो सारे के सारे क्या हो गए, प्लास्टर हो गया, सीमेंटेड होगा, कोई एक भी किसी के घर में छेद है तो अगले दिन ही सारे घर वाले टेंशन आ गए कि मच्छर एंटर करके भाई इसको। दिस इज वन फैक्टर। सेकंड फैक्टर कि जो हमारी फीडिंग हैबिट्स थी, हम क्या करते थे, अनाज खुले में पड़ा रहता था, दाने होते थे क्योंकि ग्रेडिंग होल्स, तो वो दाने हमारे इसमें थे, आंगन में पड़े रहते थे और हाउस स्पैरो आती थी, वो वहां से अपने जो है फीड देते थे। अब क्या है, जितना भी अर्बन एरिया की बात करो और इवन गांव के अंदर भी थोड़ा थोड़ा है, वो चीज इम्पैक्ट आ गया। सारा का सारा जो फूड है हमारा वो पैक्ड है पॉलीथीन में। क्या फूड हमारा क्या है, या फ्रिज में है, अगर बच गया तो बाहर तो उसको फेंक देगा। दिस वाज द सेकंड फैक्टर। और थर्ड फैक्टर अगर हम इसमें मोर डिटेल में बात, दैट इज गांव में अर्बन की छोटी बात, गांव में क्या है, भैंस होती थी, भैंस लोग जो है लाइफ स्टॉक पे ज्यादा ध्यान देते थे, तो हम दाने फेंकते रहते थे, तो दानों में कई बार है इंसेक्ट हो जाते थे। और जो हाउस स्पैरो का छोटा बच्चा है उसको अपनी प्रोटीन रिक्वायरमेंट के लिए वो क्या है, उन इंसेक्ट को खाता था। अब वो इंसेक्ट जब दाने ही नहीं खुले में है, इंसेक्ट कहाँ से आएगी, तो अल्टीमेटली उनकी पॉपुलेशन नो डाउट डिक्लाइन हुई है। मैं अपने गांव में बचपन में जब तो हमने ध्यान ही नहीं दिया, जब पीएचडी करी तब पता चला कि भी हाँ ये भी स्पीशीज होती है, इसका ध्यान देना चाहिए। जब मैंने ऑब्जर्व किया तो उनका नंबर दिखने को नहीं मिलती थी, बट अगेन उनका नंबर दोबारा से अगर गांव में जाओगे तो इट इज इंक्रीजिंग। या आप ऑब्जर्व काम करें, जो शहर का आउटस्कर्ट है और जहाँ पे पशु रखते हैं, आप वहाँ जाएंगे तो ये तो नेचुरली कहीं न कहीं हमारी जो लग्जरियस लाइफ है, और हमने अब जैसे पार्क पहले, अब पार्क क्या है, सारे के साथ ग्रीन लस ग्रीन है, उनकी क्या है, पर्टिकुलर टाइप का ग्रास है, पर्टिकुलर टाइप के एक्सोटिक प्लांट्स हैं। तो हमारे जो नेटिव प्लांट्स हैं या हैबिटेट वैसे काफी जगह होती है उनके लिए नहीं है। तो दिस इज ऑल्सो ए पॉइंट ऑफ कंसर्न एट द ग्लोबल लेवल, बट दे आर नाउ नॉट थ्रेटन्ड। ये थ्रेटन्ड स्पीशीज नहीं है, population is showing the decline, but it is not threatened species, it is still our least concerned species. Thank you.
थैंक यू वेरी मच डॉक्टर परमेश जी, ग्रेट प्रेजेंटेशन, वास्तव में बहुत ही ज़्यादा। और ये बर्ड्स का तो एक रिसेंट टॉपिक है, इस पे काफ़ी काम चल रहा है। अब मैं अनुरोध करता हूँ डॉक्टर बलजीत साठ जी, डॉक्टर गुरचरण सिंह और डॉक्टर राम सिंह बेनीवाल जी और डॉक्टर सुखेंद्र यादव जी और डॉक्टर सुशीला सांगवान और डॉक्टर निर्मला राठी, आप डॉक्टर साहब को एक मोमेंटो देकर सम्मानित करें। और डॉक्टर जितेंद्र मेहता जी प्लीज। डॉक्टर परमेश कुरुक्षेत्र यूनिवर्सिटी कुरुक्षेत्र में वेल नोन जोलॉजिस्ट है और नेशनल इंटरनेशनल लेवल पर काफ़ी प्रेजेंटेशन उन्होंने दी है, मुझे भी साथ एक दो बार जाने का अवसर मिला। थैंक यू डॉक्टर साहब फॉर सच ए नाइस प्रेजेंटेशन।
आप लोग जैसे कि अच्छा तो कुछ ज़्यादा ही वर्ड्स कहती है कि भाई हाँ रिनाउंड साइंटिस्ट, ऐसा कुछ नहीं। आई एम वन ऑफ द टीचर और ये बाई प्रोडक्ट है, ये जो रिसर्च है कि आई हैव इन्वॉल्व वेरी मच इन द टीचर एक्टिविटी ऑफ वन ऑफ मेरे जो साथी हैं सारे साथ डॉक्टर बलजीत है कि भाई अब हमने भी क्या है, हमारे पास इतना ना तो टाइम था ना हमारे पास इतने औजार थे, इंस्ट्रूमेंट थे कि मैं लैब इस्टेब्लिश करूँ और इतना, क्योंकि नेचुरल है आप प्रशासन से तो हमने एक अनदर बात कि भाई इकोलॉजी का भी जो फील्ड है वो इतना ही इंटरेस्टिंग है और इसमें आप अपनी दूसरी एक्टिविटी के साथ साथ अपना जो एकेडमिक वर्क है उसको भी आप कंटिन्यू कर सकते हैं। तो इसलिए मेरा उन साथियों से जो मानो चाहे यूनिवर्सिटी लेवल पे या कॉलेज लेवल पे जहाँ पे इंफ्रास्ट्रक्चर नहीं है या ये डरते हैं पीएचडी स्टूडेंट तो आने वाले को कराएंगे कुछ, अपने आप हो जाएगा। एक भाई आप पीएचडी स्टूडेंट को लो, 2-4 इंस्ट्रूमेंट आप खरीदो जो मैंने बेसिक से बताए, उसके बाद जितनी हेल्प जिसको साथी को चाहिए हम अवेलेबल हैं। तो इसमें कोई ज़्यादा और ये भी नहीं कि मैंने पार्ट्स में काम नहीं किया। मेरा पीएचडी का जो टॉपिक था वो टेस्ट मैनेजमेंट था, मैंने तो टोटल 0 से शुरू करके आज जो है 3 पीएचडी स्टूडेंट कंप्लीट कर चुके हैं और 6 अभी कर रहे हैं। और नॉट ओनली बट फिर मैंने कहा कि भी बटरफ्लाई में भी काम शुरू कर दो, 2-4 बच्चे उन्होंने बटरफ्लाई भी शुरू कर दिया। फिर इंडियन फ्लाइंग फॉक्स जिसको चमगादड़ करते लड़की, दूसरी एक मेन बात और भी कहता हूँ, पहले ये लगता था मुझे भी कि भाई ये तो लड़कों का काम है, लड़कियां काम फील्ड में जाएंगी, ऐसी कोई नहीं है। जितने 8 स्टूडेंट्स मेरे अंडर हैं उनमें से सिर्फ एक लड़का, 7 बाकी जो 10 स्टूडेंट हैं, तो ऐसा करने की जरूरत नहीं है। एक बार आप शुरू कीजिए और आगे सारे आपको सफलता मिलेगी। थैंक यू।
Unknowingly we missed one important award. We apologize for that. I invite Dr. Jitendra Mehta to present this award. May I invite Dr. Sahdev Singh, former Deputy Commissioner, Planning Commission, Minister of Agriculture, current Dean of College of Agriculture, Galgotia University, Greater Noida. He is being presented with Best Academician Award. I invite Manoj also for Young Achievers Award. Do we have Manoj here? microecology. He has conducted extensive research on various bioinputs being used in the natural farming practices and has also been extensively working on microbial diversity of dung from different animals. He is recipient of various prestigious awards. One of them is DAD, Indo-German Award 2003, and Raman Fellowship Indo-US 2013 for postdoctoral studies in Germany, England, and USA. Of late, in 2021, he was awarded the prestigious Swiss Pasteur Award for his contribution in the field of microbiology and sustainable agriculture. Recently, he has been conferred with Best Agricultural Scientist Award in 2024. Please give him a huge round of applause. He has trained more than 1,100 scientists, 2 lakh farmers, and 3,000 students for natural farming practices. So we are extremely delighted to have you here. His topic of presentation is Microbial Marvels, Harnessing Nature's Tiny Titans for Environmental Sustainability. So,
it's my pleasure that I am presenting my work in this college. This college is my alma mater. I did my BSc in 1995 from this college. Basically, I am thankful to Dr. Satish, Dr. Sijendra, for giving me this opportunity, this platform. And this is a very good conference, international level conference, and the basic topic is vast area of the world, that is climate change and sustainability. Climate change and sustainability, if we talk of, that is very, very important. Every organism, microorganism, plants, living animals, everything is very important for all of us because they are involved in biochemical cycling. And if there is any kind of contaminant in the environment, so that is harmful for every type of cell, that is maybe plant cell, maybe animal cell, or maybe microbial. Dr. Parnes has discussed about birds, that is the zoological aspect of biology, that is also very important. And my focus will be on microorganisms. Keening research is not being seen by naked or unaided eye. As the topic is microbial marvels, that is harnessing nature's tiny titans for environmental sustainability, my main focus will be on environment and agricultural microbiology. So there are some issues. Basically, if we see, we talk of environment, we talk of agriculture, everywhere that is being contaminated. In air, you see a lot of pollutant. In agriculture, there are a lot of residues, that is pesticides, insecticides, and high loads of synthetic chemicals that is in the air. And many problems because of mutations. So ultimately, both these issues may start with the use of these pesticides, insecticides, and contamination of agriculture and contamination of environment. And all these issues, they are related issues. Other problems, there are regarding this increasing temperature, the climate change. That is the major issue because this is going to affect all type of habitats. Basically, habitat is the natural home of all living beings. The habitat will be disturbed. That, for example, in the case of human beings, animals, birds, and microbes, everything will be disturbed. Our enzymes, they are working at a particular temperature. If that temperature is being affected, then metabolism is affected. Metabolism is affected, then you see all processes, biological processes, they are affected. If we see the temperature 37, it's okay because if temperature is high, our body is responding. Temperature is very low, our body is responding. Likewise, microorganisms. The biodiversity of microorganisms, that is also dependent on the temperature and availability of substrates. Temperature and availability of substrates both are affected by temperature, and this is the main thing, climate change. How we can overcome climate change if temperature is increasing, how the microorganisms they will affect the sustainability in coming years. So you see, the temperature is affecting the life of plants, the life of animals, and everything is being affected because the can overcome these factors, these stresses, the plant can overcome these factors if there is sustainability, or you can say if there is synergism or microclimate condition is there. We are maintaining microclimatic conditions, it's okay. We are creating natural conditions. Otherwise, if we are disturbing any one component of these, then, for example, if we use a high dose of chemical fertilizers or pesticides, if dose of chemical fertilizer is high, that is toxic, directly it kills microorganisms, other organisms growth is affected, and pesticide, insecticide, they are poisons, they are also killing. If we are using the split dose of urea and other manures, then that is also affecting, because in that case you will see plant is growing, but sometimes substrate is more. So one kind of microorganism is dominating, and another kind of microorganism that number is very less. In that case there is imbalance, and if there is any kind of imbalance, so sustainability is affected. Sustainability is the continuation of race of the earth. Everything is working in natural conditions, okay. But if there is any kind of stress, then it will directly or indirectly affect the life on earth. So we see in case of water also, volcanoes and everywhere there is kind of problem, and the conditions they are affecting the environment, and ultimately there is a shift in the biodiversity involved. Right? So environment and microbes. Main thing is we can see the trees, we can see the birds, and the farmer can see everything in his field, but he is not able to see the microorganisms. Because if you see the definition of microbes, microorganisms are the organisms which can't be seen by naked eye. If we talk of the size of any organism, any living being on earth, if it is 0.1 millimeter in diameter or less than that, we are not able to see. So those are the microorganisms: algae, fungi, bacteria, viruses, helminths. So any organism having its diameter or its size 0.1 millimeter in diameter or less than that. So main thing is, obstacle is to see these microorganisms. And these microorganisms, they are the major key players of agriculture and environment. Everywhere, if there are no microorganisms, you will see if there is any kind of wastage or any dead and decaying organic matter. So decay will not be there. If microorganisms are there, there is a succession process. So you will see desert will be converted into forest. The thing is, microorganisms, they are involved everywhere except fire. In fire, they are not able to survive because the temperature in fire is very high. Second thing is, major component of our cell is water. Water is evaporated. And third thing is, major component of biomolecules is proteins. They are denatured, and the vitamin is lost. So microorganisms, they are present except fire. In desert, you will see, even if we are keeping the curd in freeze, so after 15-20 days, you will see there is a kind of smell. Rhesia bacteria is there. It will start because it is a kind of cyclophilic bacteria. From desert also, from salty lake also, due to osmoregulation, there are adaptation, microorganisms are present there. So extremophiles, you will see everywhere. If they are present everywhere, they are playing role. What kind of role they are playing? They are harmful also. Any type of disease that is being caused by microorganisms, any type of product, you see any industry, any factory, food industry or fermentations, everywhere these microorganisms, they are playing very important role. Next. In case of agriculture, you see the number is very high. Microorganisms number, if we talk of 1 gram of soil, it contains 1 crore of bacteria, 10 million bacteria per gram of soil. If we talk of 1 kg of soil, so multiply by 1000. If we talk of 1 hectare, it's containing almost 2.24 into 10 raised to the power 6 kg of soil. That is the soil involved in agriculture in a particular area. So the number is very, very high. And these microorganisms, they are playing in every case. You see, in case of biting influence, abitic influence, suppress pathogenic effect, and increase solubility. That is the uptake of nutrients. So they are helping the plants, our plants, by 16 to 17 ways, directly or indirectly. Directly means they are giving the manures, and indirectly means they are protecting the plants from the pathogens or from the diseases. Next. The important thing is, on Earth, nearby 17 elements they are required for life. The life of plants, the life of animals, the life of microorganisms. They require 17 elements. If there is proper movement of these elements, so these elements, the life is not sustainable. We know biogeochemical cycling. The cycle of every element is important. For example, carbon. So, important thing in agriculture is organic carbon. Organic carbon, from where it comes? Ultimate source is photosynthesis. From the environment we see 0.034% volume by volume carbon dioxide is there. That is fixed by plants or dead and decaying organic matter. So carbon moves in the atmosphere, moves in the environment, in everywhere, in flora, fauna, aquatic, land. So this is moving, so that is very good thing for sustainability. If we are fixing carbon and it's not moving, or if we are burning the agro residues, it will go into the atmosphere in the form of carbon dioxide. So the thing is, to maintain the sustainability, the biogeochemical cycling is very, very important, and all the elements like carbon, they should move. If they are not moving, the formation of recalcitrant compounds is there, and these recalcitrant compounds, they are not available to the plants. If we talk of soil of Haryana, the pH is going up to 8. At high pH, most of the enzymes, they are not working. At high pH, most of the biotransformation processes, they are not occurring. So that's why our plants, they are facing the deficiency of many elements. For example, if you see zinc, so in our soils, in some soil, there is no deficiency of zinc. But plant is showing because the soil is in bound form in recalcitrant compounds, and the microorganisms are not there. They have been killed because of high dose of synthetic fertilizers and pesticides, and ultimately there is no availability of zinc to the plants. Next. So if we talk about microorganisms are just like our society. In our society we are having doctors, we are having judges, we are having drivers, we are having politicians. Every component of the society is very, very important. If there are no teachers, who will teach? If there are no doctors, so who will take care of health? Every component is important. Likewise, in microbiology, every microorganism, if we talk of different groups like bacteria, and if we talk of individual bacteria like lactobacillus, E. coli, Pseudomonas, every organism is very, very important because their habitat and substrate utilization is important. If we are having AFTES or any complex compound in our home, so you will see after 1 or 2 months, the Pseudomonas will grow because it is having megaplasmids. Any substrate that is being utilized by microorganisms. Likewise, we are having a different type of interest regarding food. Some people like butter paneer, some people like kheer, some people like pulses. So different microorganisms, their substrate is also accordingly, and all microorganisms, they are important. If one of the microorganisms is not there inside the soil, then the biogeochemical cycling will be affected, and availability of nutrients will be directly affected, and that will make the soil unfertile. Next. So these are the habitats. If specifically we talk of agricultural microbiology, they are associated with leaves, they are associated with stem, and major portion is with the roots, that is rhizobacteria. These rhizobacteria, they are very, very important. Phylobacteria with the leaf, endophytes, they are having those microorganisms which can make temporary houses with the plant structures. If there is crop, they are present, they are doing their functions. If there is no crop, even then they are present. And free living, if there is no crop, the maximum number of free living microorganisms is there. For example, azotobacter. So it's very important bacteria. Temperature is high, it will convert into cyst. That is dormant structure. Temperature is low, it will multiply. The amount of nitrogen is less in soil, then it will go for biological nitrogen fixation. Then symbiotic, some of the bacteria, they are in symbiotic relationship with the plant. For example, rhizobia, bradyrhizobia with the pulses. If you see the roots of pulses like chickpea, so you will see small nodules. Those nodules, they are containing these bacteria. The color of the nodules is pink because of the lag hemoglobin that is protecting that bacteria during the process of biological nitrogen fixation. Some microorganisms, they are of associative type like azospirillum, that is also going for biological nitrogen fixation. But the thing is, when the moisture is more, canopy in the field is very high. In that case, this bacteria comes. If we see the field of sugarcane, if we see the field of wheat in the month of February, this bacteria, its number is very, very high at that temperature, and it goes for various activities. Mycorrhiza, basically the concept is of higher roots of higher plants and fungi. So this is a combination of root with the fungi, and this is obligate combination, and this is very, very useful. If we with the roots there is fungi, mycorrhiza is there. So it is protecting plants indirectly or indirectly 17 ways. If soil concentration is high, it's protecting the plant. If moisture is less, if moisture is high, intensity of light is high, so many factors. So if mycorrhiza is there, so plant is not feeling lonely, and any kind of stress that is biting or abiting, it will not affect it. It's in. Next. So we see the scenario in the soil when the farmer starts sowing, there is sowing. The seed looks into the nearby conditions. It checks the day length, the moisture, how many hours is night, what is the content of water, is there any toxic substance, is there any heavy metal. Everything is okay, then it will start germinating. If there is any kind of limiting factor, it will not start germination. So if there is no germination, it doesn't mean seed was not good, seed was not viable. So these factors of the soil, they are also affecting the seed germination. As soon as the conditions are good, it starts germinating and it releases root exudates. That root exudate is having the food for microorganisms, that is having volatile organic compounds, and that attracts the useful bacteria. And these are the live cells. They multiply 1 into 2 into 4, 18, 16. Their number is going on, and the biomass of the root is increasing, and the microbial communities, they are also increasing. So it starts with sowing, and up to harvesting it is playing a role. After that, the death of some kind of microorganisms as per the environmental conditions starts, and the organic content is increased. So microbial flora is more, then the organic content is more, and for example, organic carbon, it goes up to 1%. So in forest soil, if we see, the organic carbon is up to 2%. In our agricultural field, the organic carbon is 0.5 or less than that. So that is a very big problem, and we have to increase. So we have to shift towards the sustainable technologies like natural farming, organic farming. Next. So these are the role how these microorganisms they are contributing, how they are helping the plants. Biological nitrogen fixation. So what is the major source of nitrogen in the for agricultural land? That is urea. If we see the process of urea formation, that is Haber-Bosch process, and that requires very high temperature, very high pressure, and catalysts like nickel. Ultimately, they are causing the pollution in the environment. And if we see these teeny creatures, that is microorganisms, rhizobia, bradyrhizobia, and this azotobacter, they are going for biological nitrogen fixation, and they do this process at room temperature. So at 37 or 30, they are going for biological nitrogen fixation. And throughout the world, if you see, the total biological nitrogen fixation is more than that the amount of urea produced worldwide by industries. So if we are using urea, so that is the nitrogen source. And these microorganisms, they convert atmospheric nitrogen into nitrate, nitrite, ammonia, available form of the nitrogen, and plant is getting nitrogen directly from the atmosphere. It is being converted into nitrate, nitrite, and then that is being utilized by plants. Then phosphate fertilization, there is no limitation of phosphorus on the every rocks, mountains, hills, they are containing, but that is the inorganic form that can't be utilized by plants. So that should be solubilized, that should be converted into phosphate, PO4. That is the plants they can go for its uptake. So phosphate fertilization is very important. These microorganisms they are doing that. Phytohormone, that is very important because it will decide when the plant will grow, and this flowering will happen, at which stage there will be harvesting. So all these processes, they are controlled by hormones, and we are talking about plants, so we are talking about phytohormones. Intermediate metabolism is directly affected by these plants. The main thing is the plants, if there is any kind of deficiency, there will be no synthesis of these plants. These plants, they give signals. If there is any kind of deficiency, they give signal to the rhizobacteria, and rhizobacteria, they will go for the synthesis of these phytohormones. Then micronutrient mobilization, and all these processes, they are helping plants directly or indirectly. Biocontrol. Next. Biocontrol, this is the process in which one organism is controlling other organism. If cat is catching mouse, it means this is biocontrol. If any bacteria is controlling another bacteria, phytopathogen, this is also biocontrol. So if there is sustainability, if we are following natural farming, what type of microorganisms they are present, there will be competition between these phytopathogens and useful bacteria. Ultimately, the useful bacteria, its number will be more because the requirement of the useful bacteria, they are less. So if we are not adding synthetic things, we are not disturbing, there is natural phenomenon, then you will see there are very less chances of coming disease. Then bacteriocins, these are the small peptides and these are antibiotic-like substances, but their molecular weight is very less. Basically, these are peptides. They control phytopathogens. There are many bacteria, especially lactic acid bacteria. They synthesize these bacteriocins, and bacteriocins, they control the disease. Then biosurfactants, these are the surface active compounds. If biosurfactant producers are there in the soil, their number is very less, but they are very important because they are producing surface active compound. Their role is to reduce the surface tension of water. You see, the small drop of water is round. If the bigger drop is there, then it is flattened because of the gravity. So this round small drop is because of the surface tension of the water. So these surfactants, basically, they reduce the surface tension. For example, if we are using soda, soap, detergents in water, then the dirt is removed easily. And if we are not using these detergents, we are washing the clothes without detergents, the dirt is not removed because these detergents, they are surface active compounds. Likewise, biosurfactants, they are surface reducing compounds. And you see the scenario in soil if there is Wapsa creation. Wapsa is a condition where there is 50% moisture and 50% air inside the soil. So that is very important for the microbial flora and for the development of plants. So in case of Wapsa formation, there are small droplets. They are round, they are having, they are containing nutrients, but that nutrient is not available to the plants because of the surface tension. If these biosurfactant producers are there, the surface tension will be reduced. The small droplets, they will spread and it will come in contact with the roots of the plants and there will be uptake of nutrients. So it is helping the plants in getting nutrients from the soil. Antibiotics, when we are getting infection of any kind of microorganism, we are taking antibiotics. Doctor is advising antibiotics to us. Likewise, these plants, they also need antibiotics. If there is any kind of attack of phytopathogen, so the signal comes, the plant is giving different kind of signals. Likewise, when we are not happy, our face is different. If we are happy, then expressions are different. Plant also is giving expression in terms of signals. So any kind of signal, light is also a signal, sound is also a signal, and smell is also a signal. So these, the smell is because of the volatile organic compounds. Our plants, they are releasing volatile organic compounds and they are being captured by phytopathogens and phytopathogens attack. If our rhizospheric soil is very enriched in microorganisms, useful microorganisms, these useful microorganisms, they produce antibiotics and that antibiotic controls the phytopathogens. ACC-deaminase, this is a kind of enzyme. It is also being produced by many microorganisms, many bacteria. If there is any kind of deficiency or any kind of stress, early maturity is there. So this early maturity of the plant is because of the ethylene, and that ethylene it may be controlled by this ACC-deaminase. That is, it is converting the inflammatory pathways towards alpha-ketobutyrate. There is no formation of ethylene. If in the environment there is no ethylene, then there will be no early maturity of the plants and it will be more. The thing is, if there are ACC-deaminase producers more in soil, so ultimately they are increasing the yield of the plants by indirect mechanism. Next. Allospheric bacteria also, they are equally important. They are having all these properties, 16 to 17 properties. They are helping in biodecomposition, they are helping in protecting bacteria from diseases, they are helping in photosynthesis process, evaporation. All processes they are being involved and these microorganisms, they are affecting the development of the plant when they are present even on the leaf of the plant. Next. So from where these microorganisms come, if we want to use more microorganisms, so as I have told, they are present everywhere. We go for isolation. We are targeting one kind of microorganism. For example, if we need biological nitrogen fixture, so we are giving specific media. We are taking samples. We go for its dilutions. We are feeding it, and we are getting very good number of colonies. And one colony, one bacteria multiplying to 2 into 4, 18, 16, 32, 64. Ultimately, many number of bacteria. In 16 hours almost, it's doubling. So we are getting more than 1 crore million, trillion bacteria if there is substrate. So that is the strategy how we see these bacteria. We are not able to see single colony, single bacteria. For example, this house is made up of bricks. We are not able to see the brick if it is, we are giving it 500 meters or more than that. But if we are making the house of bricks, we can see that house from very large also, maybe from 1 kilometer. Likewise, if there are few cells, we are not able to see them. But these cells, they are multiplying, we are supplying the food, and there is formation of colony overnight. Next day, we are counting the colony. One colony means one bacteria. So that is, and that multiply by the dilution factor, that is the strategy. So we see the photopathogenous sites, the sites where desirable microorganisms will be there. We are isolating that, we are purifying that pure culture containing only one type of cells, and we are utilizing in our experiments. Next. There are two approaches. Some bacteria we can cultivate on our given media, that is on battery plates, but many microorganisms, they can't be cultivated on this media because they are not able to know what kind of food they require. In that case, we go for metagenomic approach. In that case, we take a sample and we isolate the DNA, and on the basis of DNA, we recognize the microorganisms. So that is the metagenomic approach. And how these microorganisms number is increasing or decreasing if we are facing the problem of length of days. For example, temperature is very high and the length of day is very high. So how these microorganisms, they are fluctuating. For example, you see, you get any kind of one color, red color, increasing, decreasing, increasing. In between that kind of microorganisms, they are increasing or decreasing. They are affecting the plants directly, they are affecting the microflora also. So if there is high temperature during the month of February, last week you see early maturity in wheat is there. So such type of problems they are facing in agriculture because of change in temperature or terminal heat is there. If there are microorganisms, they are helping these plants to alleviate the stress of temperature in that case, for example in case of wheat. Next. Then we go for direct counting of microorganisms. We can see we can take any sample food material or soil samples or dung. We are talking of manure, so we take the dung of different type of animals and we see it is a good indicator. If the number of microorganisms is more, it means it is very good manure. So likewise, if we take a different type of crop sequences or rotation or crop patterns, so we compare it. If more number of microorganisms, it means that soil or that manure is very good for the agriculture. Next. This is also the number. You see, 10 raised to power 7 is 1 crore and 10 raised to power 9 is 100 crore. So 3.4 into 10 raised to power 9. So that is very, very high number of microorganisms. These microorganisms, they are the labors for the farmers. Overnight, they are doing a lot of biotransformation processes. They are having a lot of milk in our houses in the evening, and we put this culture, starter culture. In the morning, we get a lot of curd. So that milk is being converted into curd. Likewise, there are so many processes in the field, and these microorganisms, different type of microbial communities, they are working and they convert different complex compounds into simpler one, and they are the labor for the farmer. If the number is more, the fertility is more, and there are chances of sustainability. Next. Likewise, different components, if they are making any formulation, any kind of manure is there, geometry. So this is a kind of formulation which in natural farming, the farmers they are using. Total volume is 200 liters. For example, they are taking 1 drum and full of water. It is containing 10 kg of cotton. It is containing 8 liters of urine. It is containing 2 kg of jaggery or wood and 2 kg of besan and 500 grams of soil from the pond, where there is no pesticide or there is no chemical. So ultimately this bioformulation in 4 days we get geometry, and this is very good. It is containing a lot of microorganisms because microorganisms they are coming from the soil, microorganisms they are coming from the cow dung. So this is very good combination if we talk of plant protection, if we talk of plant nutrition. Plant nutrition, biological nitrogen fixer, human fertilizers, plant protection, that is antibiotic producer. Both type of microorganisms they are present in this formulation. So such type of many formulations these days are being utilized by the farmers, and that is very much required these days. Next. Then we perform these various type of properties, whether there is biological nitrogen fixation or not, whether there is an enzyme which is present or not, these microorganisms, especially bacteria. Next. Siderophore, this is a kind of compound. This is a chelating compound. It attracts the metals, and if there are contaminated sites, if we have these siderophore producers, they will chelate, they will bind all the metals. If there are heavy metals, it will attract towards it and it will bind them, so that the pathogens, they are not able to get these cofactors, and indirectly the siderophore producers, they control the disease in case of phytopathogens. Disease caused by phytopathogens to the plant. Next. Then in vitro also, if there is a kind of violation microorganism, you can see the change in color. If there is yellow color, it means that microorganism is violation microorganism. Next. Then we go for antifungal activities. There are so many fungi which phytopathogens which cause diseases in plants. So if there are bacteria which can control these fungi, there is aerosol formation. Like last photoplate you can see, there is very big zone. It means it is very good biocontrol agent. That bacteria is controlling the fungi. Next. Chitinase activity also, that is enzyme also very important. This is also chitinase is the enzyme which can degrade the cell wall of fungi, that is made up of chitin, and ultimately phytopathogens is being controlled in natural conditions. Next. Then we go for the purification and characterization of that compound. What kind of compound is being produced by microorganisms, and that is extracted and purified and can be used for commercial purposes and in agriculture. That is very important because this will control all type of pathogens. Next. Then advanced technology. Ultimately, we try to find out the different functional groups. This kind of functional groups are there present, and what kind of compound is there. Next. Next. And to see the direct effect of these microorganisms on the germination, we see. So if germination is very good, that microorganism is very good because it is acting as plant growth promoting microorganism or plant growth promoting bacteria. So different type of seeds we see for the germination. This is our farm where we are not using any kind of manure, we are not using any kind of, there is no water source also, even then the crop is very good. So we see all these conditions under natural conditions, and we isolate microorganisms and we characterize them and then compare with the conventional farming, organic farming, natural farming. There is difference between conventional farming, that is we are using chemicals and pesticides. Organic farming, we are using the inputs from the market, but in natural farming we are not using the inputs from market. The farmer is preparing all type of bioformulations at his home or in the field home. So that is reducing input cost. Next. Then as per the metagenomic approach, we see different groups like archaea, next, viruses, bacteria, fungi, the whole metagenome, whole microbiome of that particular site. What kind of microorganisms they are present and how it is different from conventional and is organic and natural farming. So the important point if we talk of sustainability, microbiology is very important component, and in case of microbiology, if any farmer is doing farming or any industry is going for industrial products or effluent treatment is there, all those processes, bioremediation, microremediation, rhizoremediation and plant protection, they require experts, then trainers. So main thing is to understand the concept. If we talk of production of any kind of produce industry or effluent treatment or natural farming, concept is very important. Then trainers, they are very important. We are producing trainers, farmer trainers. We are also doing workshops, conferences, mission, formulation, sustainability, and ultimately the purpose is poison-free agriculture. Because of this poison, a lot of problems are there. Allergies, heart attack, diabetes, you can see at very low age, thereby 10 to 20, 30, our younger ones, they are getting a lot of problems like allergies, diabetes, heart attack. Those are because of these poisons. Because these poisons, they are directly affecting our system and indirectly they are affecting the produce in the agriculture. So thank you so much if there is any kind of details. कोई क्वेश्चन है किसी के माइंड में एग्रीकल्चर से रिलेटेड, एनवायरमेंट से, मॉलिक्यूलर बायोलॉजी, कोई भी अगर कोई क्वेश्चन हो।
यूनिवर्सिटी में आप काफ़ी लंबे समय तक रहे, इस समय प्रिंसिपल साइंटिस्ट हैं चौधरी चरण सिंह यूनिवर्सिटी, हरियाणा एग्रीकल्चर यूनिवर्सिटी हिसार में, इनका डिपार्टमेंट ऑफ माइक्रोबायोलॉजी। और ऑर्गेनिक फार्मिंग, नेचुरल फार्मिंग में प्राइम मिनिस्टर का एक कमेटी है, टीम है, उसमें 5 वाइस चांसलर हैं और 2 सीनियर साइंटिस्ट हैं, एंड यू आर इन द प्राइम मिनिस्टर टीम ऑफ द 7 मेंबर्स। और प्राइम मिनिस्टर के साथ डायरेक्ट कई मीटिंग हो चुकी है। नेचुरल फार्मिंग जो मिशन है उसका सिलेबस है ऑल ओवर द इंडिया, आप उसमें काम कर रहे हैं और लगभग 40 कंट्रीज में आप विजिट कर चुके हैं। लगभग कई जगह आपका लेक्चर और रहता है, तो आज आप यहाँ आए, बहुत अच्छे इंसान है, ज़मीन से जुड़े हुए हैं, तो आप यहाँ आए, आपका बहुत बहुत धन्यवाद डॉक्टर बलजीत साहब जी। और एक हम मोमेंटो हमारी तरफ से डॉक्टर जितेंद्र मेहता जी, डॉक्टर नामा जी, जया मेहता, डॉक्टर साहब परमेश जी, डॉक्टर गुरचरण जी और मैडम सुशीला सांगवान जी, प्लीज आ जाए।
थैंक यू डॉक्टर साहब। डॉक्टर साहब मैं ज़्यादा ही पढ़ाई करती, मैं एक सिंपल आदमी हूँ, यहीं अपना गाँव उकराना के पास, और मैंने BSc यहीं से की, MSc PhD भी यहीं से की। और कभी भी कोई भी किसी तरह की प्रॉब्लम हो एग्रीकल्चर से रिलेटेड, मॉलिक्यूलर बायोलॉजी, माइक्रोबायोलॉजी, बायोटेक्नोलॉजी, बायोकेमिस्ट्री, कोई भी स्टूडेंट वो आ सकता है और अपनी यूनिवर्सिटी पास ही है। तो पर्पज़ यही है कि जब हम रिसर्च करते हैं, टीम वर्क होता है, तो किसी भी तरह की प्रॉब्लम आ रही है, चाहे वो PhD से रिलेटेड, रिसर्च से रिलेटेड, तो एनीवन इज मोस्ट वेलकम। थैंक यू डॉक्टर साहब।
सीनियर मेंबर्स हैं डॉक्टर वेद भूषण जी और राजवीर सर जी, आप भी प्लीज आ जाओ सर, कोई बात नहीं, एक फोटो में तो आ ही जाओ। डॉक्टर साहब आ रहे हैं चीज से, आ जाओ सर।
इन फलों को अपनी स्मृतियों में संजोने के लिए आपको एक स्मृति चिन्ह दिया जा रहा है। आपने कहा मैं ऐसा नहीं हूँ, ये मेरी बड़ाई की जा रही है, ऐसा नहीं है। सहारण में अक्षर को भी आगे पीछे करना पड़ता है, सराहना हो ही जाती है, और ऐसे व्यक्तियों को अवश्य होनी चाहिए जो वास्तव में धरातल से जुड़े हुए हों, अपनी भूमिका को पहचानते हों, अपने काम को बड़ी संजीदगी से करते हैं। आप उन्हीं में से एक हैं। और अब मैं मंच पर आमंत्रित करना चाहूँगा
Pankaj is here present to receive the award. Receiving for Best MSc Thesis. May I request Dr. Jitendra Mehta to express his words of gratitude.
Thank you, Dr. Biotechnology, to provide me an opportunity to express my words in front of wonderful audience. Many people have made unexpected contribution to the successful conduction of the first international conference on natural resource management and environmental sustainability for future generations. And I feel very happy to thank them. In this series, Dr. Jitendra Mehta, President, AFTES, first thankful to our chief guest, Professor Dr. Narsim Rao Vishnoi, Honorable Vice Chancellor, Guru Jambeshwar University of Science and Technology, under whose leadership this organization is working. Sir, your words are always a blessing for us. I thank the patron Dr. Deepmala, Principal, Government College Hisar, whose guidance we get at every step, through which we were able to reach this position today. And we hope that we will continue to get your guidance in future also. I express my gratitude to the organizing secretary of this conference, who provided unique support in generalizing the entire program. I warmly thank the strong pillar of the whole conference and also convener Dr. Satish Kumar, Assistant Professor, Department of Botany, Government College Hisar, and my friends also, Conference Coordinator Dr. Krishnan Singh Nama, Vice President AFTES, who supported me outright in every aspect of this conference. I am grateful from the bottom of my heart to all the four managing secretaries, co-conveners of this conference, whose point-to-point guidance we were able to easily overcome all the obstacles in this conference. I am grateful to all the members of the National and International Advisory Committee and Advisory Secretary, who fully supported us in making this program successful. I am grateful to all those highly knowledgeable speakers of this conference. On the basis of this grassroots knowledge, help to improve the knowledge of all our participants. On behalf of AFTES and Biotech, I thank our host organization, Government College Hisar, to provide us the opportunity to organize this conference. I hope that we must have lived up to the expectation, and we will continue to get the opportunity to be involved in future also. Thank you all.
Continuing with the series of awards, may I invite Amit Singh Khatri for Young Fellow Award. For this, I invite Dr. Suresh from Jat College to present the award. Sutendra sir from GCW Next in line is Vikas Tiwari. Then we have Akshay Mehta for Best Thesis Young Fellow Award. Then we have Akshay Mehta for Best Thesis Award and Young Fellow Award. It's been awarded 2 at a time. Akshay Mehta. Then we have Shivam for Young Fellow Award. Shivam Pandey for Young Professional Award. Here Shivam Pandey for Young Professional Award. Is anybody left? Is there anyone who is recipient of the award but not announced his name, his or her name?
बोटनी जी और उनको हम अवार्ड दे रहे हैं, जोरदार तालियों से अभिनंदन करें। डॉक्टर एम.एस. स्वामीनाथन अवार्ड और मैं अनुरोध करूंगा डॉक्टर परमेश, डॉक्टर बद्री सारण और डॉक्टर गुरशरण जी आप आए। सभी 2 अवार्ड के बाद सभी हमारे जितने भी यहाँ डेलीगेट्स बैठे हैं सबसे अनुरोध रहेगा कि पहले आप लंच कर लें, उसके बाद जहाँ जहाँ आपका टेक्निकल सेशन है वहाँ आप पहुँचें और 2 बजे हम टेक्निकल सेशन शुरू कर देंगे। कोशिश करेंगे कि आपको 3:30 या 4 तक हम फ्री कर दें आज इस प्रोग्राम से। मैडम अगले अवार्ड।
I invite Sudha Shai for Young Professional Award. Sudha Shai. Next is Preeti Jha for Young Professional Award. We have Sudha Shai for Young Professional. Then we have Preeti Jha Now all of you are invited for a lunch. We have a hall in the back of the auditorium.
हाँ जी, जिसे कैमरा दिख रहा है उसकी फोटो आ जाएगी. Okay.
And my dear colleagues and participants from different parts of the country, a very, very warm welcome to all of you in the technical session of this first international conference on natural resource management and environmental sustainability for future generations. In this technical session, first of all, I would like to introduce you to the chair of the session, Dr. Kamla Malik, who is a senior scientist of microbiology department from HAU, Hisar, Haryana. She has research experience of 20 years and her area of research are biofuel, fermentation, waste management and nanotechnology. She had 6 projects funded from RKB by Rashtriya Krishi Vikas Yojana, ICAR and state government of worth rupees 10 crores. Officially, she has visited 5 countries, that is USA, UK, Italy, Finland and Sweden. The list of her Best Oral Presentation Awards, Best Post Presentation Awards in national and international conferences of her research papers and certificates of appreciation for outstanding contribution in research area is very long. But I surely have to inform you that she has received appreciation letter under international collaborative project entitled Assessment of Agri-Biomass from Agroclimatic Zone of Haryana from LUT University, Finland, and also received letter of appreciation for remarkable work in research and devotion to duty from former worthy Vice Chancellor CCS HAU Hisar in 2020. She has received travel grant from Department of Science and Technology, Panchkula, Haryana, to attend international conference on energy from biomass and waste in 2018, organized by IWWE, University of Padova and Venice, Italy. She did 3 months training in Texas, A&M University, College Station, USA, under International Training Program, NAHDP IDP project in 2021-2022. She had publication of number 200. She has 200 publications. Department of Microbiology, CCSHAU, Hisar, editorial member of University Journal, CCSHAU, Hisar. And we are highly honored to have such an eminent, internationally known scientist among us. I welcome you, Dr. Kamla Malik. I welcome you, Dr. Rajkumar sir also. He is co-chairing Dr. Kamla Malik. Now I would like to invite Dr. Suresh for his presentation. Thank you very much. Dr. Suresh, please.
Thank you, madam. Good afternoon, ladies and gentlemen. Myself, Dr. Suresh Kumar, Associate Professor in the Head of Department of Zoology, Biotechnology. Today I am going to present my research paper, The Impact of the Delta Methane on the endocrine gland sodium, potassium and calcium, magnesium, ATPase in freshwater fish, microplastics fossils. As you know, the different types of the insecticides present at organochlorine, organophosphate, carbamate, pyrethroids. Today we are going to discuss about the pyrethroids. These are the synthetic pyrethroids which are easily degradable and the less toxic to the mammals. But it is very harmful and toxic to the aquatic animals. As you saw, this deltamethrin is used in the agriculture, used household pest controls and the other uses. So largely these are used in the different countries. If we see the data from the 90, the Argentina, they have increased the 815% times more pesticide used as compared to the India. India is very going to the developing country and use the heavy amount of the pesticides. But the other 20 countries, they are using more than India the pesticides per year. The next please. Next. The pesticide cycles. If we use the pesticides in the agriculture, after some time due to the rainwaters and the seepage in the soils, they reach into the water bodies and they cause the toxic effect on the aquatic animals, the mainly affected organism in the freshwater fish. Next please. All of you know the excessive use of the pesticides not only the health hazards, it also causes the environmental impact assessment. So today we are going to discuss about the effect of the deltamethrin on the freshwater fish. Next please. Deltamethrin pyrethroids, synthetic pyrethroids, one of the most widely used active ingredient in the world to pest control or work effectively at the low rate of the concentration. Next please. These are the cyanides group present on the synthetic pyrethroid group, therefore it is a heavy toxicity to the aquatic animals. Next please. These are the material or methods. First of all, we procure the deltamethrin from the market. We are generally using the agriculture. After next please. After that, we carry the some of the freshwater fishes from the natural resources, water, and implement in the laboratory in normal conditions. After that, the physical chemical property of the water, these are the pH, the normal temperatures, and that means the freshwater fish is easily acclimatized. After the acclimatization of the 15th day, we conducted a lethal test, concentration test, actually 96 hours, actually the 50% test animals will be done. That is the 0.52 milligrams per liter. We divided into two different concentration, 1/3 and the 1/6. The 1/3 is The lethal concentration is 0.17 and the 0.08 milligrams on which we conducted the experiment. Next slide, please. Next, please. The endocrine glands. In case of the freshwater species, there are the three types of the glands which regulate the calcium and the sodium, potassium, calcium, magnesium, ATPase in that one. The first is the ultimobranchial gland, prolactin cells, and the corpuscles of the species. Next, please. The calcium, magnesium, ATPase activity we have conducted the different types of the methods, include methods. Next, please. Sir. Inorganic phosphate. Next one. Now we come to the result and the discussions. Whereas we conducted the three group of the test animals, one is the control, second is the low concentration of the delta-methylene, the second is the higher concentration of the delta-methylene for the 30 days. These are known as the chronic exposure to the test animals. Next, please. Next. These are the normal structure of the ultimobranchial glands. The ultimobranchial glands is responsible for the calcium homeostasis in the freshwater species, and these are present near to the gills. These are the normal structure. Next. Next. After the exposure to the lower concentration, there is the mild changes in the endocrine system. Next, please. These are the severe affectors, and there is the necrosis and the regulations take place, which we see under the compound microscope. After the study, the histopathology into the electron microscope, next week we can see the severe changes at the higher concentration. Next week, these are the structures of the normals at the ultimobranchial gland at the electron microscope. You can see here the different types of the cell organelles, mitochondria, cell vectors, etc. But at the higher concentrations, these are next week. At the higher concentration, you see these are the less of the mitochondria, and you have seen the necrosis take place. Next slide, please. These are the result ultimobranchial gland ultimately led the severe changes at the higher concentration. Next, please. Next, please. Corpuscles of stages. These are the second types of the endocrine glands which are present on the kidney of the freshwater species. Next, please. These are the movement structure. When we expose these cells, the corpuscle of stages under the lower concentration, there is the mild changes. Next, please. At the higher concentration, there is the severe changes, the necrosis and the cell degeneration take place. Next, please. Corpuscles of stages at the higher concentrations, there show the severe changes were observed, increased granulation, maturation, and the degeneration of the cell membrane. Next, please. Next, please. Prolactin cells. These are the third essential endocrine glands which are present in the freshwater species for the regulation of the calcium homeostasis. These are the normal structures. Next, please. Next. Prolactin cells, so when we expose at the lower concentrations, there is the severe changes. Next, please. Next, please. These are the ultrastructures under the electron microscope. So we have seen the different types of the cell organelles. Next, please. Next. Prolactin cells. So the no marked histological changes was observed at the lower concentration, when subcellular level decrease in mitochondria number, endoplasmic reticulum, secretory fluid was disappear at the higher concentration. Next, please. these are the membrane bound enzymes. So they changes the concentration of the receptor. In this graph, you show the calcium ion at the higher concentration, say the muscles and the gills. These are the severely effective at the higher concentration. Next please. Next please. Sir, next. Conclusions at the end, we come to the conclusion that the delta-methylene synthetic pyrethroid is highly toxic to the fish. It interacts with the physiological processes with the changes in endocrine glands and the biochemical parameters. Significant changes observed in endocrine glands and the inhibitions in the outer of the brain is the most effective due to the cell membranes ruptures in the brains, muscles, gills, and intestines. All the activities they are the highly effective at the higher concentrations. Next please. The level of sodium and potassium decrease maximum in intestine and kill exposure at the higher concentrations, but the lower concentration significant was only in the intestines. Next please. The enzyme activity was elevated in the gill at the lower concentrations due to the excitement at the initials. They are responsible to the pesticides. Second, the level intestine brain skin were most effective tissue of the fish. Next please. The level of calcium decrease in intestine, muscles, gills, brains and the kidney. Next please. Ultimately, we can say the pesticides which are used in the agriculture and the households, they are not only effective with the mammals, but these are the highly toxic to the aquatic animals. Thank you. Also, they regulate the calcium homeostasis in the
lecture of Sri Prasad Dishta, PhD study from Nepal is going on. First of all, we will hear that, and then we will continue this technical session. Thank you very much.
One of the component of this 6 years back. But these things has international contact is has been started over in this culture, and because this has been a tradition of this culture for the last so many years. So we have another festival, particularly in India, we call Dasera, here we say Dasera. So this is one of the biggest festival. There, during that, before this festival, you can see the photographs here in my slides that maize for wheat as well as barley, they are shown, the seeds are shown in the pot culture. So we have pot culture, we make some potting methods like we mix soil, clay as well as sand, and we make a potting mixture. Then here we put all the seeds, and after 10 days, when it gets germinated, and we put the sunlight, we put it in the dark, so it becomes white color or yellowish color. And that is in Tita, put it in Pasera, and this shows that so we put our elders will give blessings to the younger son. So this is one of the biggest festival of Nepal, and in that festival we use symbols as pigments, as photographers to make those plants. So these are some of the things which I just wanted to highlight. But before I finish it, I just wanted to record soil is considered very much small. The second one was this is an animal, and we consider it as a god. Soil is second to God. So the concept of viewing this is already blended in the culture since long time. So because I say that the culture is very much related to some of the cultures, like if we see that the culture of even in the Indonesia, Japan, China, they have the same type of culture, they say that everything, I say that soil is life, because life comes from soil. So if there is life, the last form of life is soil again, because when it dies, again becomes soil. So soil is life is soil. So the first form of life was finished is the soil. And even when we say that even the Christianity, they say that the first human being was Adam is soil, and the second one is life. So soil and life goes together. That means the three things, ethical consciousness, creation, and spirit, and other is when we consider all those things, then we say that it should be promoted, and the culture should be culture should be preserved. Otherwise, if the culture goes, you lose your identity. So most of the English has come out from the culture itself. So that is why promoting during Nepal is promoting culture. This is the final slides. I just want to share this. So
continue the session. If you want to sit over here. Okay. Now I request Dr. Ravi Khan, Associate Professor in the Department of Zoology, HAU, Hisar, deliver his talk on the stage. Thank you.
Deliver my talk, antimicrobial resistance, a growing serious threat in aquatic bodies in Haryana. Next please. Next please. I am working on that aquaculture. You know the aquaculture industry contributes an aquatic sources for food nationwide as well as the worldwide. And India is the second last country in the fish producing after the China. Intensive and semi-intensive agriculture practices, especially in the agriculture, to produce the large fish stock and use of a good or ample amounts of antimicrobials used to become the animal disease free. As the consequences, the antimicrobial resistance comes into the existence and the pathogens from horizontal as well as the vertical transfer from the one species to the another, and it becomes a havoc of our society. So I, along with my team, working on the isolation of the various pathogens from the aquatic bodies in Haryana especially, and to find out the antimicrobial properties of herbs, especially the essential oils from indigenous area. And antibiotic resistance quickly spreads among the aquatic microbial communities, then reaching to the human pathogenic bacteria, making the use of antibiotics for human health way, especially after the COVID-19 era. Then the emergence and spread of antimicrobial resistance from the unrestricted use of the antimicrobial used in the livestock feed of the various contributing factors. Next please. Next please. What is AMR? We will talk about the anything that may be antibiotic, antiviral, antifungal and antiparasitic, which make the pathogen or organism make them resistant, and it is burden to whole world. More than 7 lakh people die every year due to the AMR, and main cause is the overuse and misuse of the antimicrobials in the daily life. Drug-resistant infections are more complicated to treat one or to cause disability, prolonged illness, prolonged hospitalization, and heavy bills. And WHO says that AMR among the top 10 global public threats in present era, and I am saying that after the use of agrochemicals, unjudiciously the use of antibiotics is also the second most worst problem of this era. Then AMR, according to WHO, AMR is the capital of world recognized to the India, or you can say that WHO recognized India as AMR capital of the world, and more than 70% of superbugs belong to the E. coli, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa were resistant to the most frequently wide broad spectrum antibiotics. And it is reported that colistin, that is the last resort of antimicrobial, and Acinetobacter baumannii is also found that it is not working properly, specifically in the hospitals of pneumonia patients. It was studied that ICMR in 2024, more than 95,000 bacterial culture, 24.7% of E. coli, 18% of Klebsiella, and 13% of Acinetobacter baumannii were multi-drug resistant. Next is, while India needs to curb antibiotics use, you can say this flow chart that since 2000 and 2015, the antibiotic use specifically in the cephalosporins and broad spectrum penicillin, it is tremendously used in our society. In India, bacterial disease is the burden of the highest in the world, and large population is suffering from the various diseases like diabetes, kidney disease, heart disease, cancer, which make the people more prone to the disease. and 40% of population is malnourished and which add on the risk of high infections, and more multi-drug resistant bacteria are being identified day by day. Next please. This is the few articles which showing depicting the data that India is on the high on the antibiotic uses after China, and alarming statistics show that 75% of Indians were thoroughly studied and they were using antibiotics basically for the cold and flu. Next. Next please. Burden of antibiotics in the neonates also. 1 million Indian children are dying at the age of 4 months or 4 weeks. Every year because of the several elements which occur during the prenatal stages. Next please. This is the methodology which was conducted by our team. First, isolation and characterization of the bacterial isolates from the collected water bodies of Haryana. This was the routine process of sample collection, serialization technique, bacterial culture and their identification, and then the evaluation of antimicrobial activity of essential oils against the bacterial isolates which were found to be multi-drug resistant in the laboratory conditions. Next. This shows that the inhibitory effect of antibiotics against the bacteria and several bacteria found in the Haryana water bodies was resistant to all the three concentrations of the ofloxacin and ampicillin, which are the broad spectrum most usually antibiotics in our medical treatment, and few pathogens were found resistant at all concentrations against the cefixime. Next. Four bacterial isolates were found to resistant at cefixime at 25%, and one pathogen that is usually found resistant to cefixime at 25 concentration, and similar pattern was also observed in the study. Next please. This shows the inhibitory effect of various essential oils which were used against the isolated bacteria of microorganisms from the water bodies, and found that they were found very effective in the different concentrations in the in vitro conditions. Next. Based on various multiple biochemical tests, Haryana, Pseudomonas aeruginosa, Vibrio cholerae, Enterococcus, Streptococcus species, Staphylococcus species were identified and found more prevalent in the collected water samples. Bacteria isolates were analyzed for antimicrobial susceptibility against the 5 major antibiotics, and it was found that these were found resistant against the cefixime and ofloxacin and ampicillin, which were commonly used in our medical practices. And in another study, it was reported that 30% of multi-drug resistant isolates of Haryana species are of isolated microbes reported in the village in and around Hisar. And considering the studies, it was found that herbal extract against the MDR bacterial isolate influence their management. Next please. These are the publications from our lab. Next. Next please. So what action was taken? The global effort must be made to cease antimicrobial overuse in aquaculture and encourage the stakeholders to adopt the disease prevention measures. And the National Action Plan was launched in India in 2017 by Union Ministry of Health and Family Welfare under the objectives improving the awareness, enhancing surveillance measurements, strengthening infection prevention and control, R&D, promoting investment and facilitating collaborative activities to control the antimicrobial resistance. Next, please. What are the alternatives for the antibiotics? First, vaccination. Next, we are working on my lab, that is bacteriophage therapy. Next is the botanicals, probiotic food and antimicrobial peptides. Next, please. Thank you. Judicious use of antimicrobials help us to control the multi-resistance bacteria in our society. Thank you. Any questions, please?
Now I request Shiva Pandey, who is a PhD scholar in Haryana. Shiva Pandey.
To all the committee members, my topic of presentation is effect of indigenous herbs on growth parameters of common grass, which challenge me Pheromonas side of the infection. Basically, it was my PG research work. So next slide, please. We all know that agriculture is an emerging and increasing sector and contributing very much in food sector and also the country's GDP. So there are various types of parameters, pathogens which are affecting agriculture in brackish water, fresh water and also saline water. So in this research, we are going to discuss about one of most emerging pathogen, Aeromonas hydrophila, which is basically found in freshwater fishes and causing motile Aeromonas septicemia disease in fishes. So next slide, please. In the context of treatment of aquaculture disease, we are generally using different types of antibiotics and different types of harmful chemicals which are affecting our fishes as well as human consumption also. So in this research, I am going to replace the treatments of harmful chemicals with the substitution of herbal extracts. Next please. Next. So study area is College of Physical Science, Hisar. Next. And this is technical program of work where this is the experimental species is common carp, Japanese carpio. Challenge pathogen is Aeromonas hydrophila. There was 4 treatment and 3 replications, and breeding rate is 4% of body weight. In both of 4 treatments, I have 1 treatment as a control diet. Treatment 2 as 5% of neem extract feed, and treatment 3 is 5% of tulsi extract feed, and treatment 4 is combination of 5% neem and tulsi extract. The duration of experiment is 90 days, and all the fishes were contained with 150 liter of tubs with 10 animals in per tub. Next, these are some photos. Next, treatment description, basic diet, and some of the treatment were supplemented with herbal extracts. Next, these are preparation of diet. Next thing, preparation of herbal extract. Next. I observed 5 of the growth parameters. One is feed conversion ratio, specific growth rate, and percentage increment in length, percentage increment in weight, survivability. Next. The preparation of bacterial injection. Next. Next is builder. Next. These are some pathogenic observations after the challenging of animals. Next. In treatment, with 30 days of feeding with the immunized food, we are giving injection of Aeromonas hydrophila 1 into 10 to power with 1 into 10 power 7 CFU and 100 microliters of intramuscular injection when injected per challenging. So after 30 days, the growth parameters, after 60 days and after 90 days, the growth parameters were increased. So first growth parameter is resulting is feed conversion ratio, which is showing least in 1.95 and highest value is 6.35 in T4 treatment. FCR is the parameter where the least value is very efficient. So next, this is the ratio, graphical representation. Next, Then a specific growth rate of common carp with herbal extract is found in T4 treatment with 5% combination of protein and gluten. Least value found in 30 days in treatment T4. Highest value, and least value is also found in T4 in 60 days, 0.44. Next. These are the next. Then percentage incremental length. The value of percentage incremental length is found higher in treatment T2 with supplemented of 5% T, and the least value of this treatment is found in T4, 1.51. Next. The last parameter is weight gain percentage. They are found highest value with T4 treatment for 90 days, and the lowest value found in control 30 days. Next. And the last one parameter is survivability. So survivability found at the end of how many experimental animals were harvested. So the largest quantity of experimental animals were harvested in T4 treatment combination of 5% D and QC extract. The least is found in T1 control treatment. Next thing. So here was the conclusion. I am concluding with that point. In this experiment, we are using the herbal treatments as prevention and also for treatment of the Haryana hydrophilic disease, which is causing infections. So we observe that when we are giving challenge to Haryana hydrophilic infection, then the fishes stop their feeding and also the normal behavior which they are doing every day. And the other parameters we are studying that hematocrit and all the hematological parameters related to immunity. So all the growth parameters were increased after the challenging of pathogen, and when we are continuing the feed of experimental diets, so the recovery rate is high. So with that point, I am including that if we are using the herbal treatment instead of harmful antibiotics and harmful chemicals, then it will be very good, it will be very useful and also very efficient for the environmental and also for the farmer benefit. It was very useful. Thank you so much. Yes. Aromalas bacteria.
Gurjan Singh, please come on the stage. और डॉक्टर निर्मला राठी और डॉक्टर सुशीला सांगवान जी दोनों ऑर्गेनाइजिंग सेक्रेटरी हैं। आपसे मेरा रिक्वेस्ट है कि एक मोमेंटो और एक सर्टिफिकेट देकर आप सम्मानित करें। डॉक्टर राजपाल आप ऐड करेंगे। डॉक्टर गुरचरण सिंह। डॉक्टर सुरेंद्र यादव को देकर सम्मानित करें। डॉक्टर ज्योति और डॉक्टर राजकुमार। जो पर्सन है यहाँ जो कर रहे हैं, क्योंकि सबसे ज़्यादा स्ट्रेंथ यही है, और मेरा थोड़ा अनुरोध भी रहेगा कि आप 5 मिनट से कम टाइम मिले। डॉक्टर कमला मलिक जी, आप मंच पर आए प्लीज। का थोड़ा ध्यान रखें ताकि सभी टाइम पे अपना जा सकें। हमारे सेशन चेयर हरियाणा एग्रीकल्चर यूनिवर्सिटी हिसार से Thank you all of you. Dr. Srika. डॉक्टर जितेंद्र मेहता जी एक अवार्ड के लिए कैंडिडेट है, हम देख लेंगे, वैसे तो इनका है अवार्ड बाद में भिजवा देंगे। एक फोटो डॉक्टर श्रीका। राजपाल जी इनका अवार्ड वाला वो देख ले और एक बार कोई भी दे दे सर्टिफिकेट। क्योंकि कुछ ऐसे कैंडिडेट हैं जिन्होंने ऑनलाइन अवार्ड के लिए अप्लाई किया था, हमने सिर्फ ऑनलाइन सॉर्ट आउट, ऑफलाइन सॉर्ट आउट किए थे। जो ऑनलाइन के थे वो कुछ कैंडिडेट आ गए हैं, इसलिए उनके अवार्ड हमने सॉर्ट आउट नहीं किए, हमारे पास हैं लेकिन वो अलग रखे हुए हैं। सभी के सर्टिफिकेट उनके मेल पे जाएंगे, विद बारकोड जाएंगे और बारकोड जब आप स्कैन करेंगे तो आपका सर्टिफिकेट ही ओपन होगा, वो आपके बाद में आएंगे प्रेजेंटेशन के और ये आपके अटेंडेंस के अकॉर्डिंगली ही देंगे, सबको नहीं मिलेंगे। जिनकी जहाँ भी अटेंडेंस है उन्हीं को ही मिल पाएंगे, सबको नहीं मिल पाएंगे। थैंक यू।
Now I request Sujita, PhD scholar from Hisar.
Professors and dear delegates, I am Sujita. I am here to present some part of my research work, and title of the research is methanolic extraction of bioactive compounds from fruit peel waste and then further synthesis of silver nanoparticles for agriculture application. The content of my presentation is firstly introduction, how the waste is generated and how we are responsible for that. The second one is the research methodology that I use in my research. Then third one is the results that are qualitative and quantitative. The lastly conclusion. The firstly you can see that according to the Food Safety and Standard Authority of the India, in 2022 and 2023, there are the 350.87 million ton production of fruit and vegetable in horticulture. But out of these, there are the 68.7 million ton of the food waste annually in our kitchens. Means 50 kg per person we are responsible for that. And here you can see the list of the waste. Grain is 12%, meat is 12%, dairy products are 17%, and lignocellulose like rice, wheat are 20%, and the lastly fruit and vegetable are 28%, which are higher than others. It means 1/3 of all production, that is 350.87 million ton, the 1/3 of that we are wasting annually. To keep in mind these all things, my research is depend on the waste management, how we can manage the waste that are produced in the horticulture from fruit and vegetable. But mainly I focus on the fruit peel. Mainly I use banana peel, sweet lemon peel, water chestnut peel, orange peel that I use in my research. Here you can see the research methodology that I use. Firstly, the peel waste from the fruit, treat it by reducing the particle size or by using the acid or alkaline treatment. After the treatment, I use two methods, conventional and non-conventional method. In conventional method, I use soft extraction method, and in non-conventional method, I use ultrasound assisted extraction method. After the extraction method, the crude extract, during the centrifugation, I use the crude extract for further analysis. The analysis of biological assay like total phenolic content, total flavonoid contents on the basis of the UV visible spectro and FTIR. On the other side, I use the crude extract for the preparation of the nanoparticles by using silver nitrate. I use 0.01 molar silver nitrate, and the ratio of the silver nitrate and the extract that I prepared during this research in ratio of 1:2. After that, the solution of the silver nitrate and the extract incubate for at 80 degrees Celsius till the slurry changes to dark brown, and after that the nanoparticles are further analyzed on the basis of the FTIR, and inhibit or reduce the yield of the crop, and antioxidant activity is very useful for our body or human, like radioactive oxygen species are very responsible for the stress that are the main cause of the heart disease in recent era. Firstly, the qualitative assay that depend on the UV visible spectrum. Firstly, the determination of the total phenolic content. Out of the 5 fruit peel based like banana, sweet lemon, water chestnut, orange peel and grapefruit, the sweet lemon peel and the banana peel showing the high content of the phenols. But there is the optimization. I used 50%, 60%, 70%, 80%, 90% and 100% solvent. Here I use methanol as a solvent. But in the case of the 70% methanol, it's showing the higher phenolic content as compared to the pure methanol. Now here you can see the total flavonoid contents. We can compare the phenolic content and flavonoid contents. You can see that in the sweet lemon peel, the second graph showing the higher flavonoid content, but the flavonoid content is low as compared to the phenolic content. Same result are here that the 70% methanol showing high flavonoid content as compared to the pure methanol. On the basis of all the means, the sweet lemon peel and banana peel showing the high activity like high microbial activity, high antioxidant activity, high phenolic contents and high flavonoids. Then we use these two, sweet lemon peel and banana peel, for the further analysis. What is the reason behind their activity, how they are responsible for such type of the activity. Then we use the FTIR to determine that how many types of the peaks are present in their extract. Here you can see that the peak are between 400 to 4000. They are the higher peak but not sharp in the case of the sweet lemon peel. But in the case of the banana peel, the peak are sharp but less as compared to the sweet lemon peel. Okay. Here you can see the frequency, means the peak that are showing in the previous graph, and the specific peak having the specific bond which are responsible for specific type of the stretching during the formation of any type of the crude extract and any type of the particles which are further use these extracts for making that specific material or compound or value added products. Now after the extraction, by using conventional and non-conventional method, the non-conventional method is showing the high efficiency for total phenolic content or total flavonoids or further like biologic acid like antimicrobial, antioxidant. To keep in mind that I use the non-conventional extraction method extract for the preparation of the nanoparticles by using silver nitrate. Here you can see that silver nitrate formation by using 0.01 molar solution of the silver nitrate, and the ratio is 1:2. Then after the formation of the nanoparticles, these nanoparticles are further determined on the basis of UV visible. There is the two graphs, that one is the sweet lemon peel and second one is the banana peels. In the case of the sweet lemon peel, the percentage of the transmittance is lesser as compared to the banana peels. But in the case of the sweet lemon peel, the frequency or the wavelength of the extract or nanoparticles are higher. But in the case of the banana peels, there is a one peak that's showing on the 400 nanometer. Now I use these nanoparticles for the germination of the wheat seeds. I use for concentration. First one is the zero means control, and second one is the 50 microgram per ml. Third one is the 100 microgram, and the fourth and fifth are in between like 25 microgram per ml and 75 microgram per ml. But out of these five, there are the 50 microgram per ml concentration of the nanoparticles that are prepared by using sweet lemon peel and banana peels. The 50 microgram per ml silver nanoparticles are more efficient for the germination of the wheat in the lab condition. Here you can see the germination of the wheat seeds in the concentration of the 50 microgram per ml nanoparticles from sweet lemon peels, and in the case of the banana peels. But they have the difference. In the case of the sweet lemon peels, the germination efficiency is 95%, But in the case of the banana peels, the germination efficiency is nearly 90 to 92%. It means the sweet lemon peels having the high content of the phenols, flavonoids, which are responsible for the further germination of the crop. Now I conclude my research. We are using the source of nutrients, the ignorance by the stakeholders. There are the many things that are responsible for the agri-food waste. But if there are agri-food waste, then how we analyze or how we use these waste for the further production of the value-added products. There is the method, sustainable options that we can use in labs or at the pilot levels on the industry level, like bioconversion, by using valorization methods like non-conventional or green extraction methods, because green extraction method having the high efficiency, low energy consuming and low time consuming method, and we can sustainable valorization or that can increase the circular economy, and then integrated bioremediation of these waste can be further responsible for the zero waste, and we can convert waste to the well in our environment. These are the publications which are mainly depend on the waste management that are published by me. Thank you so much.
Professors, delegates and the judging committee and my fellow co-scholars. I joined the session in between of afternoon, and since then I am listening a word very much, that is herbal content. So either we are moving for bacterial treatment towards the herbal treatment, and in nanotechnology we are moving towards the green synthesis. And in earlier two presentations we also read about the phenolic content of the food waste. So here my topic is zinc activated neem-based biochar and activated green solution for water pollution. I am Vijay Yadav, and I am presenting and I am doing my PhD under the supervision of Dr. Santosh Dugal. Next, my PhD is basically on nanofertilizer, but while I was working on nanofertilizer, I synthesized zinc oxide nanoparticles with the help of neem extract. And many of my fellow co-scholars are also using plant extract, some are using food extract, as we see in the earlier presentation. But when we extract the phenolic component or the activated component from that food waste or neem particle or any plant extract, that filter matter which is there on the filter paper, that is discarded. After the extract preparation, the powder of leaf, that is waste. So I tried to convert that leaf powder into the biochar, and further I modified that biochar with the help of zinc for the removal of, I am doing this research forward now in my lab for the removal of naphthalene, which is a prominent pesticide in agricultural waste. So here are three components in my presentation. One is biochar, second is neem-free, and third is zinc oxide. Biochar is basically a bioresource subjected to the pyrolysis in the absence of oxygen. We convert the matter into the carbon material, and which is commonly used for the fertilizing material. And second is neem-free. I specifically choose neem because neem is very much, I choose neem in my research because neem has antibacterial properties. So in this work, the neem powder is waste of my that part, which I further use. And third is zinc oxide. I use specifically zinc oxide metal because of its efficiency, and second it is easy and cost effective, and the synthesis is very easy. Next. I reduced the biochar on the nano size because nanotechnology has many benefits, because it is applicable in a lot of sectors, and because of its smaller size, the nanoparticles have more active sites to combine for the pollutants. So I specifically reduced the biochar on the nano size. Next, methodology. Neem leaf powder was first washed thoroughly with the distilled water and dried at 35 degrees Celsius. Then this dried leaf powder was kept in the muffle furnace for the pyrolysis at 550 degrees Celsius, which is an easy process to convert it into a biochar. And then it is grounded and sealed with a fine seal to make it homogeneous. And this is again dried at 85 degrees Celsius for 35 hours in hot air oven. Next, this prepared biochar is further functionalized with the help of zinc, nanoparticles. So I took the zinc sulfate of 0.1 molar and it is mixed with biochar 0.5 gram for 15 minutes, and the whole solution is titrated against the KOH for maintaining the pH and reduction in the size. So the process is repeated until the titration reached up to the mark of 100 ml, and the synthesized product was further filtered at the filter paper of size number 42, and it is oven dried. And both the component biochar and zinc modified biochar are calcinated for more active site or more porous nature at 350 degrees Celsius for 30 minutes using the muffle funnel and stored further. Next. These are the results by which I confirm the synthesis of the product. So this is the FTI showing the peak of the biochar only. So the peak at the 3417, these are because of the presence of hydroxyl group, and along the peak in near about 16 to 1618 shows the presence of carboxyl group, and there is only single peak at 874. So there is no matter, only the functional group carboxyl, alkyl and hydroxyl groups are present. Next. This is the FTIR of zinc nanoparticle loaded biochar, and it has prominent peaks in the range of 400 to 700, which shows the presence of zinc metal. Next. Further, I took the synthesized particles for the preparation, for the characterization of XRD. So XRD. Next. Next. The peak. That's a fact. These peaks show that the particle size of initial biochar is 40, and it is not that much crystalline in nature, but the zinc loaded biochar, the particle size which I calculated by the Schaefer's equation, that is 19.29, there is a almost half reduction in size, and the peaks are more prominent, which show that it is the crystalline in nature. And crystalline in nature means it has a solid structure and structure not in irregular shape. Next I go for the zeta potential. There is a shift in the zeta potential towards the negative side, which means my product with zinc is more stable rather than the biochar only. Next. Further, I am doing the application for the removal of naphthalene, so I hope we will meet somewhere else in some other conference with the application results. Thank you so much. Actually, ma'am, I already mentioned that I am working on nano fertilizer, which are made up of zinc and neem extract. So my
Hisar, and I'm continuing my PhD under Dr. Asha Gupta and for supervision of Rashiram Bishnoi sir.
Due to the availability of its whole genome sequence, we are able to study its biochemical and molecular pathways that are responsible for characterizing the major mal constituents. Next. So, Hordeum vulgare is a monocotyledonous diploid seagull crop with a genome size of 5.3 gigabase pair, and the selection of barley for specific end-use application is determined by its beta-glucan. Beta-glucan is a major non-cellulosic component that is responsible for the mal quality assessment. So, we are in the current study, we are trying to study the genes involved in the beta-glucan biosynthesis. So, in this study, we are trying to focus the in silico approach of two major candidate genes involved in beta-glucan biosynthesis, that is the CSLF gene and calosynthase gene. CSLF, the beta-glucan biosynthesis is mainly regulated by the presence of UDP glucose cellulose glucosyl transferase, that is CSLF6. It belongs to cellulose synthase like subfamily gene 6. After the literature analysis, we confirmed that the CSLF6 is the major candidate gene involved in the beta-glucan biosynthesis. So, this is the biosynthetic pathway of 4 major quality attributes that are regulating the mal quality of barley, that is the beta-glucan, starch, sucrose and fructose. In this study, we are focusing on beta-glucan. So, as you are seeing in the pathway, that the CSLF6 gene, by utilizing on the substrate D-fructose, acts and generate involved in the biosynthesis of 1,4-beta-glucan, and the other candidate gene calosynthase by acting on the substrate D-fructose involved in the biosynthesis of 1,3-beta-glucan. These are the preference of barley amongst other cereal crop, as after threshing and harvesting, the husk of the barley remain adhered to the catalopsis and that aid in the filtration process and make it more suitable cereal crops amongst other mal sorts such as rye, sorghum and wheat. Next, sir. So, we are in this study, we are targeting the molecular modeling and docking analysis to confirm through in silico approach the candidate genes and the efficiency of different transcripts. So, for a better understanding of the structure and substrate specificity, refined 3-dimensional structure of CSLF6 and CH3A were constructed, and the molecular modeling of both UDP glucose cellulose glucosyl transferase, that is CSLF6, and the calosynthase was performed using the 5A2 software that is used to construct the 3-dimensional structure of a protein, and the further validation of the most refined models and Ramachandran plot calculations was accomplished using the project server, and for better understanding of the structure and substrate specificity, refined 3D structures were constructed and the molecular analysis was done using the server CBDoc, and the 3D structure were analyzed through the biology. So, these are the 3-dimensional structures of different transcripts of cellulose synthase like subfamily genes. These are the 9 different transcripts we obtained after the transcriptome sequencing of 4 barley genotypes, 2 indigenous and 2 exotic genotypes. Next, these are the 3D molecular structures of the calosynthase 3 gene that is obtained after the pilot research. Next, so, after analysis and after the 3D structural analysis, we come to know that the transcript with the gene ID HR07H3.3 that is involved in UDP glucose cellulose synthase subfamily with a catalytic site. This is the most abundant and most efficient transcript amongst all 9 transcripts, and this is confirmed through the in silico analysis, and through molecular docking we come to know that the ligand was observed to interact with specific amino acids including lysine, isoleucine, leucine, phenylalanine, isoleucine, phenylalanine, methylene, etc. with a binding score of -10.4 in case of CSLF6, and in case of CS3A the efficient binding score was -8.1. These are the 3D structures obtained after the CB2 structure. These structures show the binding of substrate with its ligand. Next, these are the different structures of different transcripts CSLF6 line structures. Next. So these are the different CB2 3D models obtained for different transcript of CSLF6 and calosynthesis. Now, for the flow check analysis, we go for the software SAFE 6.32. From the SAFE analysis, we come to know that the transcript with 0.9 transcript in case of CSLF6 shows maximum allowed regions 92.7 in the favored region in case of CSLF6, and in case of CS3A, the maximum allowed regions was 9.5%. These are the Ramachandran plot of different transcript of CSLF6 and CS3A. Next. These are the transcript obtained for the CSF3A. 15 transcript were checked for CS3A. Next. This is the summary of in silico analysis, that from the same value we come to know that the transcript at 074.9 with 92.7% allowed amino acids in the allowed region, and on the basis of the efficient binding scores we come to know that the transcript 0.3 has the maximum efficiency amongst all transcripts. Next. These are the data of CS3A. In this, the transcript 15 shows maximum efficiency based on its SAFE value, and the transcript 4 on the basis of efficient binding score. Next. These are the scope and relevance of our study. So, basically we are trying to optimize the transcript efficiency among different transcripts using the in silico approaches. So, the identification and the characterization of candidate gene will provide new insight into the regulatory aspect of the pathways, which in turn work as a cue to enhance or lower the production of the grain beta glucan, and we are trying to develop or possibly development of the low grain beta glucan germ class with superior indigenous mal development. Reference. Thank you. Any suggestions? We are
requested to complete their presentation within 5 minutes.
हमारे पोस्ट प्रेजेंटेशन के जजेस HUC हैं, इनको जाना भी है, काफ़ी सुबह से आए हुए हैं, तो मैं इनवाइट करता हूँ डॉक्टर शुभम लांबा जी को, प्लीज कम ऑन द स्टेज। डॉक्टर निर्मला राठी, डॉक्टर सुशीला सांगवान, डॉक्टर दीपिका और डॉक्टर मुकेश आंगड़ा, आप आ जाओ चाहो। और डॉक्टर साहब को एक मोमेंटो और एक सर्टिफिकेट। राजपाल जी, पोस्टर प्रेजेंटेशन का रिजल्ट हमारे पास आ गया है, ग्रुप में भी हम शेयर कर देंगे। और जजेस पोस्टर प्रेजेंटेशन के आपके सामने हैं। फर्स्ट प्राइज है पारुल शर्मा, यहाँ प्रेजेंट हो तो ठीक है। ये ये बाद में मिलेगा सर्टिफिकेट आपको। और सेकंड है अनुराधा और अंशु, दो है सेकंड, अनुराधा एंड अंशु है यहाँ पे प्रेजेंट। एंड थर्ड है शिवम एंड आशा रानी। तो ये पोस्टर प्रेजेंटेशन का है। In room number 72, two best presentation have been selected. One is Neeta from IC College of Community CCS HA Hisar. और सेकंड पोजीशन है नीतू CCS HA हिसार। यहाँ दोनों हैं प्रेजेंट। हाँ गुड शाबाश। आपके भी जो सर्टिफिकेट हैं वो आपकी मेल पे आ जाएंगे। ग्रुप फोटो आपका होगा एक बार आप ले लेंगे। हमारे यूनिवर्सिटी ऑफ साइंस एंड टेक्नोलॉजी से डॉक्टर सुरेश कुमार मित्तल जी काफी लंबे समय से वहाँ जजमेंट दे रहे हैं और काफी गर्मी में वहाँ बैठकर लंबे टाइम तक काम करना पड़ा, इसके लिए सॉरी सर। और मैं आपको स्टेज पर आमंत्रित करता हूँ, डॉक्टर श्री सिंगला जी आप साथ आएंगे और डॉक्टर जितेंद्र मेहता जी आपका सबका मोमेंटो, सर्टिफिकेट और अवार्ड। डॉक्टर सुरेश कुमार मित्तल जी। डॉक्टर सुरेश कुमार मित्तल जी मार्केटिंग एंड फिनांस के प्रोफेसर हैं, स्कूल ऑफ बिजनेस मैनेजमेंट। और आज सेशन चेयर किया है सर ने, और कौन सी दो पोजीशन रही वो भी प्लीज थोड़ा बता के जाएं हमें। डॉक्टर साहब आ रहे हैं साथ में, आप भी आ जाओ सर। आप एक फोटो थोड़ा रह गया, सॉरी, आज एक फोटो दोबारा करवा लेते हैं, आ जाओ।
टेक्निकल सेशन जो रूम नंबर 105 में हुए उसमें जो बेस्ट प्रेजेंटेशन है, फर्स्ट है रिया कुंडू, PhD स्कॉलर HA हिसार से। सेकंड जो बेस्ट प्रेजेंटेशन है वो प्रैक्टिस है, करिश्मा PhD स्कॉलर है, और डॉक्टर अशोक असिस्टेंट प्रोफेसर है, लवली प्रोफेसर यूनिवर्सिटी।
रूम नंबर 43, इनका भी हमारे पास रिजल्ट आ गया है, डॉक्टर सतेंद्र यादव जी और डॉक्टर सुमन चौधरी। फर्स्ट है मिस सरिता फ्रॉम गवर्नमेंट कॉलेज हिसार, केमिस्ट्री डिपार्टमेंट। second presentation, Alpna Gupta, Assistant Professor, HC College for Women, Hisar.
Now I request Nirudima to continue her presentation.
Good afternoon to all present here. Today's topic of concern is broad-induced shifting bacterial community dynamics, understanding their ecological resilience. Drought is one of the major abiotic stresses that can reduce the crop yield. Along with this, it can also induce some changes in the soil ecosystem also. Like it can increase the soil moisture level, allow their physiochemical properties such as pH and nutrient availability. The rhizobacterium that are present within the rhizosphere, they are most important in encouraging the plant under this type of stresses. They are involved in producing their types of plant promoting properties like inhaling uric acid, acetic acid, cytokinin and indole acetic acid. These drought-tolerant bacterial symbionts that are present in the rhizosphere in the root nodules, they may play a crucial role in enhancing the plant resilience to water limitation by facilitating the nutrient acquisition, modulating the hormone levels like indole acetic acid, which helps in the growth promotion of the roots, and inducing the systemic resistance against various kinds of pathogens. So the current study deals with the isolation of these drought-tolerant bacteria. The 10 bacterial isolates, they were screened for drought-tolerant 50-day soil growth, having different concentration of polyethylene glycol. Polyethylene glycol induced drought stress under the lab conditions. It is a non-permeable solute. The base were further incubated at 30 degrees Celsius with agitation rate of 120 rpm for 96 hours and turbidity was measured at 600 nanometer. The positive drought-tolerant strain were screened for exopolysaccharide production, and further time-course profiling of this strain were evaluated for proline, pH, electrical conductivity, and optical density. This first graph showing the osmotic stress of bacterial isolates. All the bacterial isolates having slow growth rate, but under non-stress conditions, their growth is normal. The absorbance at 600 nanometer recorded at 0 R is between 0.09 to 0.25, but after 96 hours of the incubation, the optical density increased to 1.6, that indicates the growth of only 3 bacterial isolates out of 10. Hence, they are drought tolerant. This is another graph showing the optical density values in non-stress, moderate stress, and severe stress conditions. The value of optical density are 1.12 in growth without pack, 5.63 with 20% pack is having moderate stress, and 30% pack is severe stress with having OD of 0.27. Now these drought-tolerant strains were further proceed for exopolysaccharide production. Aniline blue is the primary screening method for this exopolysaccharide production, having specific concentration of sucrose on them, and the blue color colonies indicates the positive color of the positive estimation of the exopolysaccharide production. The positive blue color colonies of drought-tolerant bacteria were further screened for quantified for exopolysaccharide production. These tubes showing the blood area having exopolysaccharide. This is the time course analysis of exopolysaccharide bacteria in 96 hours, and at 120 hours maximum exopolysaccharide was 15 microgram per liter. 42 isolates show maximum exopolysaccharide production. This is the time course analysis of electrical conductivity, OD, pH, and proline. The EC values remain constant throughout the time period. The OD value increases for 24 to 120 hours. The pH value increases at 40 AR, 8 hour and 96 hours respectively, while proline value decreases at 96 hours. This is the correlation analysis from these isolates can be used for further field studies and for further lab study to evaluate their production in their dot cells. Thank you. Yes, ma'am, I have used aniline in which this aniline binds to beta-1,3 glucanamides of exopolysaccharide. This indicates the production. I can also use them in industries, in port area studies or in field studies against various kind of stresses like salinity stress, drought stress, and heavy metal stresses to remediate these kind of pollution from the soil. Thank you.
Thank you. Now I request Sugandha, PhD scholar from Hisar.
I'm here to present some part of my research work, that is the assessment of groundwater quality for irrigation purposes in the Jalalabad block of Firozpur under Punjab. So as we know that India being the largest user of groundwater and it extracts 230 km per cube of groundwater annually, which is about 1/4 of the global total. And Punjab is an agriculture dominating Indian state, having 83% of its total geographical area under intensive cropping, and 98% of intensely cultivated area is irrigated. And it is among the Indian states to adopt the green revolution technologies and have the highest consumption rate of fertilizers. The agriculture practices have shifted from the conventional sustainable organic farming practices to the unsustainable modern practices, which affect the groundwater at a very higher rate, and by which the groundwater pollution is causing day by day as very serious concerns. And my area of study was Jalalabad Tehsil under the Fazilpur district in the Punjab, and here I collected 50 groundwater samples from 50 different villages, and here in Fazilpur district of Punjab, agriculture is one of the major source of groundwater pollution. In order to protect the health of rural population, it becomes more important to identify the quality of water which is used as irrigation and drinking purposes. And there is no systematic study on groundwater quality of Jalalabad Tehsil has been done previously. So my research study is on the assessment of the groundwater quality of Fazilpur district. So here I collected 50 groundwater samples from different villages, and I done the total 16 physicochemical parameters of water and 9 irrigational water quality parameters that are based in exchange index, mutagenesis index, residual sodium carbonate, sodium percentage, Kelly's index, magnesium hazard, permeability index, residual sodium bicarbonate. This is the results. In the 50 different samples, the base exchange index range was from the 0.841 to 835 milliequivalent per liter with average value of 14.22, which shows that sample belong to carbonate type water, and metagenesis index shows that its range vary from 1.0115 to 36 milliequivalent per liter with an average value of 14.76 milliequivalent per liter. And sodium adsorption ratio, in which 54% groundwater samples of present study area were found under excellent categories and 26% under good and 66% in unsuitable categories. And in RSC, 84 groundwater samples during determination were found unsuitable for irrigation, and the sodium percentage also was under the unsuitable categories, and percent Na, in which 50% groundwater sample was found unsuitable for irrigation purpose. And in carry ratio, 90% of groundwater samples were found unsuitable for crop. And in magnesium hazard, 68% samples were found fit for soil and irrigation purposes. And in permeability index, 80% samples of present study area was found bad and unfit for crops, where RSBC values stand from 2.39 to 18.9 0.08 millicurie per liter with mean value of 7.262 millicurie per liter. It shows that the water in the Hisar district is unfit for irrigation purposes because due to the excessive use of fertilizers and pesticides. That was the result of fire. Because this area was unexplored and no educational studies was done in this area, that's why I choose that. Ma'am, in this particular area there was no honey industry because it is a sub-block of Hisar district and the excessive use of fertilizers done in that area, that's why I choose that area. No ma'am. I just use physicochemical parameters and irrigational water quality parameters. Thank you.
Now I request Priyanka from Achieve. Priyanka, please.
Very good evening to everyone. Today I am here to present oral presentation on the topic of impact of particles on the wide variety of food life, that is Drosophila melanogaster. As we all know, that nanotechnology is the branch of science that deals with the manipulation of materials at nano scale, and it describes the synthesis, manipulation of and characterization of materials and structures that have one or more dimensions at nanoscale. The fundamental component of the nanotechnology is nanoparticles. The nanoparticles are various today nanoparticles are used in many fields of science, which was the toxic effect on the development and the physiology of the non-target organisms and the environment. Nanoparticles can be widely used in agriculture also in the form of nanofertilizers, nanopesticides, and nanoherbicides. So, for my study, I study the impact of zinc oxide nanoparticles on the Drosophila because Drosophila is the stable
Dr. Kamala.
Good evening to all of you. It's a wonderful session and I think all are present very well. And congratulations to everyone. So finally we have decided which one is the first and second. So first is going to Miss Sujita and second is Sugan, and this second is divided into two. First is Sugan and second is Priyanka. Second position is divided into two persons. Okay. So first Sujita will come on the stage.
Professor Rajkumar, you are also requested to come on.
Sugan and Priyanka, please.
आ जाओ
आ जाओ
डॉक्टर रविकांत जी आ जाओ। तुमने पता एडवाइस बिना पीएचडी तो नहीं होती। आ जाओ।
रविकांत सर इधर आएंगे।
एडवाइस का गाइडेंस तो जरूरी है। फर्स्ट एक्सपीरियंस था, मैं आपको क्लियर बता दूं, और इसमें हो सकता है कमियां भी रही हो काफ़ी। कॉलेज लेवल पे हमारा पहला प्रयास था और जो एक पूरी टीम थी AFTES की और वाइटल बायोटेक पब्लिकेशन की। एक आदमी ऐसा है जो लगातार 10 दिन से 12-12, 13-13 घंटे कंप्यूटर के ऊपर जिन्होंने काम किया है, प्रोफेसर कृष्णेंद्र सिंह नामा, और वो आगे 10 दिन आगे और काम करने वाले हैं। तो मैं उनको स्टेज पर बुलाना चाहता हूँ। वाइस प्रेसिडेंट हैं AFTES के, और आपके जो बार कोड्स लगे हुए ई सर्टिफिकेट सभी की मेल पे जाएंगे और ग्रुप में पूरा का पूरा वो आ जाएगा। बार कोड इसलिए हम बात कर रहे हैं कि ऑथेंटिकेटेड होंगे, बार कोड को आप स्कैन करेंगे आपका सर्टिफिकेट ओपन हो जाएगा। एंड रिटर्न तो आप कभी भी कहीं से भी बनवा सकते हो, उसका कोई बात मायने नहीं है। आपकी मेल पे आएंगे और पार्टिसिपेशन के भी आपकी मेल पे भी आ जाएंगे, और कोशिश करेंगे कि हार्ड कॉपी आपके एड्रेस पे पहुंच जाए। ठीक है, तो मैं इनको बुलाना चाहता हूँ इनके साथ में। हमारे AFTES के प्रेसिडेंट और इस पूरे कार्यक्रम जो पूरा ऑर्गेनाइज हम कर रहे हैं, इसके सारे कोऑर्डिनेटर हैं डॉक्टर जितेंद्र मेहता जी, और इन्होंने कहा कि आप प्रोग्राम करिए, फाइनेंस की कोई दिक्कत नहीं है, कम्प्लीटली हम 100% इसको स्पॉन्सर करते हैं, कॉलेज का कोई 1 रुपया हमारा खर्च नहीं हुआ। तो इसके लिए जोरदार तालियां डॉक्टर जितेंद्र मेहता जी के लिए। और पूरा टेक्निकल वर्क इन्होंने किया है, पेमेंट रिसोर्स हमने यहाँ दी है, बाकी सारा टेक्निकल काम किया है। तो मैं चाहूँगा कि इनको हमारे जो डिपार्टमेंट्स है बॉटनी, जोलॉजी और बायोटेक्नोलॉजी के वो मेंबर्स आ जाएं और एक मोमेंट देकर इनको सम्मानित करें। और ये राम भी हैं और रतन भी हैं, बहुत अच्छा इन्होंने काम किया है, तो इनको भी हम सम्मानित कर रहे हैं एक मोमेंटों के साथ। जो प्रेजेंटेशन में जहाँ किसी भी सेशन में फर्स्ट, सेकंड या थर्ड रहे, सेलेक्ट हुए हैं, वो सारे एक बार आ जाएंगे, उनकी फोटो कराएंगे, उनको सम्मानित भी करेंगे। आ जाओ मैडम, आ जाओ। जो भी है, जो भी जिसकी प्रेजेंटेशन में पोजीशन रही है वो सारे इकट्ठे आ जाएं जिनकी प्रेजेंटेशन में पोजीशन रही है। डॉक्टर सरिता को अलग से बुला देंगे। डॉक्टर सरिता ने प्रेजेंटेशन में तो अवार्ड जीता ही है, सुबह से शाम तक काम भी बहुत किया है इन्होंने, अलग अलग कमेटी में जो इन्होंने अपना वर्क भी किया है, तो उसके लिए भी इनका बहुत बहुत धन्यवाद। और डॉक्टर अशोक जो है वो जालंधर से आए हैं, लवली प्रोफेशनल यूनिवर्सिटी से, और रात को 11:30 बजे पहुंचे। आप सोच सकते हैं कि रात को 11:30 बजे मैं फोन अटेंड कर रहा हूँ, जी कहाँ रुकूं। तो ये एक लंबा प्रोसेस होता है जब भी आप कॉन्फ्रेंस करते हैं। बढ़िया, इनका रुकने का भी प्रबंध हमने करवाया। न्यूज कवरेज नहीं हुई तो हमारा कुछ नहीं होना, फिर न्यूज कवरेज बहुत बढ़िया करवाती है मैडम, थैंक यू मैडम। मास कम्युनिकेशन और मीडिया के इंचार्ज हैं, और आज प्रेजेंटेशन भी रही है मैडम की।
रिक्वेस्ट होगा प्लीज स्टेज पर ही रुकें, क्योंकि जितनी भी हर्डल्स, जितने भी बैरियर्स, जितनी भी प्रॉब्लम्स थी वो आपने झेली है, और हमारे को काफ़ी सपोर्ट किया, और पूरे टीम को कैसे ऑर्गेनाइज करके और इस कॉन्फ्रेंस को ऑर्गेनाइज करवाना है। नो डाउट कुछ कमियां नहीं है, एंड फ्रॉम AFTES बायोटेक की तरफ से मैं और हम सब उसके लिए हम सॉरी फील करते हैं। और आगे आने वाली एक्टिविटीज़ में हम लोग कन्फर्मली इसको बहुत अपडेट करेंगे। बट मैं सतीश सर से गुजारिश करूँगा, प्लीज़ सर आप इन फ्रंट ऑफ द कैमरा हैं, ताकि हम एक, वैसे तो औपचारिकता है कि आपको सम्मान, अदरवाइज आप इससे बड़े सम्मान के अधिकारी हैं। प्लीज़ सर। और मैं सभी से गुजारिश करूँगा, प्लीज़ अपनी करतल ध्वनि के साथ में एक बार
और हमारे ऑर्गेनाइजिंग सेक्रेटरी दोनों इकट्ठे आ जाएं। दोनों तीनों डिपार्टमेंट ने बहुत काम किया है। बायोटेक से मैडम आ जाओ, आप बायोटेक को रिप्रेजेंट कर दो। आ जाओ कोई बात नहीं। डॉक्टर साहब थोड़ा बिजी थे आज। कोई स्टूडेंट रह गया हो जो मतलब इनको ना मिला हो कोई सर्टिफिकेट, तो अभी एक फोटो करवा देंगे हम, इसके बाद फिर हम राष्ट्र कोई भी स्टूडेंट जो रह गया हो इकट्ठे आ जाए। उसके बाद हम ग्रुप फोटो करवाएंगे राष्ट्रगान के तुरंत बाद, और राष्ट्रगान होगा, उसके बाद ग्रुप फोटो होगी, और उसके बाद आपका धन्यवाद। ये जो मैं कर रहा हूँ उसको धन्यवाद मान लें आप। पहले ग्रुप फोटो करवा लें। ये दोस्त हैं, इन कुछ पार्टिसिपेशन की हार्ड कॉपी आई है, कुछ रह गई हैं। वो हार्ड कॉपी जो रहेंगी वो आप बाई पोस्ट आपके पास पहुंच जाएगी। तो सभी पार्टिसिपेंट का और एचयू के कई प्रोफेसर का हमने इसमें सहयोग लिया, उनका भी धन्यवाद है। जीजेयू से भी प्रोफेसर आए हैं अलग अलग सेशन को चेयर करने, उनका भी मैं यहाँ से धन्यवाद व्यक्त करता हूँ। प्रिंसिपल मैडम का धन्यवाद व्यक्त करता हूँ, और जो भी मैनेजमेंट पे लगे हुए हैं हमारे तीनों डिपार्टमेंट बॉटनी, जूलॉजी और बायोटेक्नोलॉजी, उसके सभी स्टाफ मेंबर का मैं धन्यवादी हूँ एज ए कन्वीनर। और डॉक्टर दुष्यंत ने भी बहुत काम किया आज, इनका भी विशेष रूप से जो है आज मॉर्निंग से लगे हुए हैं। एक बार सम्मान पट्टिका से डॉक्टर दुष्यंत का भी आप मान सम्मान कर दें। सर थैंक यू। डॉक्टर बडोनी जो है वो उत्तराखंड से आए हुए हैं, उनके साथी उत्तराखंड से आए हुए हैं। आपका बहुत बहुत धन्यवाद सर, और सभी का जिन्होंने एक्टिवली इसमें पार्टिसिपेट किया है, सुबह से बैठे हुए हैं। तो इस कॉन्फ्रेंस के संयोजक होने के नाते मैं सबका दिल से आभार व्यक्त करता हूँ। इस पूरे इवेंट को आपने सक्सेसफुल किया, और मेरा निवेदन, एक बार ग्रुप फोटो के लिए आप आ जाएं, कुछ जो स्टूडेंट हैं वो आगे आ जाएं, टीचर्स ऊपर आ जाएं, और एक ग्रुप फोटो आप सभी की हो जाएगी। जो हमारी PDF रहेगी उसकी बैक साइड का ये फोटो रहेगा। फ्रंट